So the story behind this project is that I have a rare genetic disease of the muscles, as you might know, and unfortunately without a cure. So I specialized in human genetics to understand the mechanisms of pathogenicity of rare diseases, including mine. And now I'm curious about how to treat these diseases. So by definition, rare diseases are disorders that affect fewer than 2,000 people. And also, although these disorders are often chronic, debilitating, and also life-limiting, there are few effective drugs, and only 5% of them have an approved FDA drug. And this is largely because pharmaceutical industries are unable to recover the costs associated with R&D and marketing of these drugs. So there are many challenges in drug design in rare diseases. This includes scientific challenges like limited understanding of disease mechanism, small patient population, genetic and phenotypic diversity, which is the hallmark of rare diseases, and also financial and market challenges, regulatory challenges, challenges also in drug development, like target identification and validation, also ethical and social challenges, like access and equity. Indeed, high treatment costs may prevent access for many patients, especially in low-income settings. And by addressing these challenges through advanced technology, policy changes, and collaborative efforts, the field of rare disease drug discovery might be more efficient and impactful. Bioinformatics and artificial intelligence have revolutionized drug repurposing and drug discovery by leveraging omic data like genomics, transcriptomics, proteomics, metabolomics, and these approaches integrate large-scale datasets with advanced computational tools to uncover therapeutic opportunities. And through this workshop, we will have some examples of applications of such innovative approaches in the field of drug design and repurposing. So what do we know about rare genetic diseases in Tunisia? Our lab is a reference center for rare diseases, and it's contributing to their better characterization on the molecular level. Through a meta-analysis of published data, we have estimated the spectrum to approximately 600 genetic diseases, of which 60% of them are autosomal recessive, and this is mainly due to the high rates of consanguinity in our population. And we are currently collecting data on pharmaceutical and effective drugs to know how many of them have an efficient drug, and we expect that only a few of them, of course, have such efficient drugs. Through our meta-analysis, we have noticed the co-occurrence of two or more genetic diseases in one individual or different members of the same family. And we call it this phenomenon comorbidity, and we have centralized the information on more than 40 comorbid association. And this phenomenon makes not only the diagnosis challenging, but also the genetic counseling and the therapeutic management. So in order to contribute to a better management of rare diseases in Tunisia, we have founded a startup called Inerfa, along with my team member, Dr. Nisreen Mizzi. And we are developing a platform called Premedit that aims to accelerate the diagnosis of rare genetic diseases and optimize their treatments. So Premedit integrates clinical, genetic, epidemiological, and pharmaceutical data on these diseases and leverages bioinformatics and AI for in silico drug discovery and repurposing. And we hope that this workshop will be a platform for insightful collaboration in this journey of making a future without the constraints of genetic diseases. So this event is funded by AGIA, that stands for Arab German Young Academy of Sciences and Humanities. And I am honored to be a part of such academy because it transformed me and opened for me opportunities to build collaborations and especially to know wonderful people. So AGIA has one mission, is that to connect excellent scholars from 23 countries from Germany and the Arab world and bring them together to face challenges and develop solutions through research cooperation. AGIA has been established in 2013 as the first bilateral young academy worldwide, and AGIA members are early career scholars from all fields of natural, life, social, and technical sciences, humanities, and even arts. And AGIA members are selected in a highly competitive selection process for a 5-year membership. So AGIA has many goals and objectives that includes interdisciplinary research, but also capacity building and promotion of high potential, research-oriented policy advice, and network building and scientific cooperation. AGIA organizes 2 annual conferences, one in autumn in one Arab country, and the last one has occurred last week in Tunis, and the second one in Germany in spring. And during these annual conferences, AGIA members discuss and decide on future research projects and joint initiatives, and also they elect the steering committee. So these annual conferences are organized in collaboration with cooperation partners in the Arab world and Germany. AGIA promotes research through different formats and working groups. So among the research formats, we have the working group projects, the tandem and alumni projects, the idea competition and research mobility, and we have many working groups, including Arab and German education, health and society, and innovation. So there are many benefits of AGIA membership, like implementation of innovative Arab-German research projects, conducting also research at the interface of science and society, also increase the professional visibility and mobility, and especially also to receive project funding and administrative support. There are many criteria of eligibility to be an AGIA member, and I highly encourage you to learn more about them and about AGIA by visiting the AGIA website and also to follow AGIA on their page on social media. So thank you for your attention and please enjoy the workshop.
Thank you, Dr. Lila, for this nice presentation. And hello everyone, welcome again to this session. So I'm Muslim Messi, I will be moderating this session. And let's start by introducing our first speaker, Dr. Radwan Al-Hidi. So Dr. Radwan Al-Hidi is a doctor in pharmacy, and he is an assistant professor in pharmaceutical chemistry at the Faculty of Pharmacy of Monastir and the National Agency of Medicines and Health Producers. So with a solid foundation in pharmaceutical science, Dr. Radwan leverages his academic and professional expertise to advance pharmaceutical research and education while contributing to the regulation of medicine and health products. So, Dr. Radwan, the floor is yours.
So, good morning, everyone. So, first of all, I would like to thank Dr. Ben Ramdan and the whole team of the Pasteur Institute for the invitation. And I would also like to apologize on behalf of Dr. Noor Ben Fatma, who could not join us today. So, in today's lecture, we will try to have a quick or brief oversight over some basics and fundamentals about drug discovery and drug repurposing. we will start with an introduction to place drug discovery and drug repurposing into the pharmaceutical life cycle. Then we will talk about drug discovery, drug repurposing, and finish with a conclusion. So when trying to define drug discovery and drug repurposing, we first need to have a look to the pharmaceutical life cycle of a given drug. So the pharmaceutical life cycle usually has 5 stages, which begins with pre-discovery, which centers around basic research, clinical observations, which aim to study molecular mechanisms involved in the disease and the identification of potential targets. Then, once we understand the physiopathology of a disease, we then move to stages 2 and 3, which center around drug discovery and preclinical studies. In drug discovery and preclinical studies, we will identify potential therapeutic agents modulating the selected target. We will study their ADMET, which stands for pharmacokinetic properties, as well as their pharmacodynamic properties. And we can use tools such as in silico, in vitro, and in vivo tools. Then we have the 4th stage, which is the clinical trials, which can be divided into 3 phases. Phase 1, which is conducted on healthy individuals, phase 2, which is conducted on a smaller sample of patients, and finally phase 3, which is conducted on a much larger scale of patients. And in this clinical trial stage, we will try evaluating some basic properties of the drug candidates, such as the pharmacokinetic, the dosage, the efficacy, and the toxicity. Then after phase 3, we then pass to the regulatory aspect of a drug life cycle, which is the registration and approval by national regulatory authorities. And then we have phase 4, which is the monitoring of mainly adverse effects of drugs after in post-marketing. So when we try to define drug development, drug discovery, and drug repurposing, we find that drug development speaks mostly to the complete process, bringing newly discovered drugs to the market, starting from early pre-discovery all the way through phase 4. So the drug development englobes the totality of the pharmaceutical life cycle. Drug discovery, on the other hand, falls within the medical, biotechnological, and pharmacological fields, and is the process of going from a clinical observation of a given disease to the discovery of a potential drug candidate. Drug repurposing, on the other hand, also referred to as drug repositioning or drug reprofiling, is an approach that identifies new clinical applications for approved or investigational drugs outside the compound's originally intended use. So drug discovery will be mostly found in fundamental research stages, such as which are stage 1, 2, and 3, whereas drug repurposing will mostly be found in later development stages, mainly the clinical phase 3B, more exactly, and the clinical phase 4. So the main problematic with the research and development is mainly about costs. And this graph, you can look at the expenditures for research and development in pharmaceutical companies from 1995 to 2009, and we can see that over a decade we have had a 3-fold time increase in the drug development, going from $15 billion to $45 billion in the US alone. And the latest estimates put the cost of developing a new drug to more than $1 billion, with a mean of $2.6 billion. And 30% of these costs are solely dedicated to drug discovery, which highlights the importance of drug discovery optimization. Now, moving on to drug discovery. So as per its definition of identifying and optimizing therapeutic compounds, the drug discovery process requires many skills and the use of various advanced technological platforms, which are often a combination of computational and experimental approaches. Historically speaking, drug discovery can be divided into two main categories, which are traditional drug discovery and rational drug design. For traditional drug discovery, 5 main approaches are usually described, which are random screening of synthetic compounds, chemical and natural products, trial and error, in which we usually screen the activity of parts of plants or natural products, ethnopharmacology, which relies on the observation of the effects of indigenous drugs, serendipity, which is the accidental discovery of a therapeutic effect of a given compound, with the most prominent example being the discovery of penicillins, and finally classical pharmacology, which is also known as a function-based approach or a phenotypic drug discovery, in which different compounds with that specific targets are studied to measure complex responses in vivo, such as the prevention of experimentally induced seizures in mice. And then traditional drug discovery remains limited since it's kind of a shot in the dark. So scientists and researchers have come with a more systematic approach to treat the drug discovery paradigm, which is the rational drug design. Rational drug design, also called reverse pharmacology, begins with a hypothesis that modulation of a specific biological target may have therapeutic value, and that the target is capable of binding to a small molecule, potentially modulating its activity. Depending on the set of data obtained during the preliminary research, we can distinguish two types of rational drug design, which are structure-based drug design and ligand-based drug design. So for structure-based drug design or direct drug design, it relies on the knowledge of the three-dimensional structure of the biological target, which can be obtained through experimental methods such as X-ray crystallography and NMR spectroscopy. And if the experimental structure is not known, it's possible to create a homology model from a related protein. So structure-based drug design two approaches are the most used in structure-based drug design, which are virtual screening and de novo drug design. For virtual screening, we identify a new ligand for a given receptor by searching large databases of 3D structures of small molecules to find those fitting the binding pocket of the receptor, and it's mainly studied by docking. For the de novo drug design, ligand molecules are built within the constraints of the binding pocket by assembling small pieces, which can be either atoms or molecular fragments. And the key advantage of such method is that we can obtain novel structures, hence the name de novo drug design. But overall, to conduct a structure-based drug design study, 4 phases are usually necessary, which are the preparation of protein structures, in which we will remove unnecessary chains, equilibrate hydrogen charges, and correct pH values. The identification of a binding site, where we will identify the XYZ coordinates of the binding pocket of the target, the preparation of ligands, where we will prepare the 3D structure of the small molecule, and of course we will minimize its energy, add hydrogens, and correct its pH. And finally, docking and scoring function, which reflect the affinity of the ligand for its target, and we can further the experiments by studying the nature of the interaction between the small molecule and its target. So for ligand-based drug design, which can also be referred to as indirect drug design, ligand-based drug design is mainly used in case of inexistent or insufficient data about the three-dimensional structure of the desired target. And three main approaches are usually used. We have the similarity modeling, which relies on the knowledge of other molecules that bind to the biological target of interest. So if 2 molecules are similar structurally, they could behave the same. The pharmacophore modeling, in which molecules that already bind and assert an effect on the primary target may be used to derive a pharmacophore, which is the the main scaffold responsible for the activity, that defines the minimum necessary structural characteristics a molecule must possess in order to bind to the target and have an effect. And finally, an alternative method could be the QSAR study, which stands for Quantitative Structure-Activity Relationship, in which a correlation between calculated properties of molecules and their experimentally determined biological activity may be derived. Ligand-based drug design may also be used to build a model of the biological target based on the knowledge of what binds to it, and the resulting model may be used to design new molecules. However, regardless of the drug design method used, both structure-based drug design and ligand-based drug design lead to the identification of a set of bioactive compounds, which are known as primary hits, and which are molecules that have the potential to interact with the chosen biological target. These compounds are then taken through further screening to confirm their activity, potency, and possibly to gain an early measure of the specificity for the target. These compounds are then known as secondary or confirmed hits. Later on, these hits become lead compounds and are defined as a new chemical entity that could potentially be developed into a new drug by optimizing their beneficial effect and minimizing their side effects. Lead optimization generally consists in adding or suppressing molecular fragments or functions, but also replacing pre-existing fragments or functions by bioisosteres in order to modulate activity, pharmacokinetic properties, and safety profile. All the tools cited earlier, such as virtual screening, de novo drug design, computational modeling, pharmacophore modeling, can be used for drug optimization. Now, to have a look at some drugs that were approved and that are derived from a rational drug design methodology, we can look at the case of imatinib, which is an oral targeted therapy medication that inhibits multiple tyrosine kinases and is used for the treatment of a rare form of chronic myeloid leukemia. So in the late 50s, the Philadelphia chromosome was discovered, and its role in chronic myeloid leukemia was identified. Later on in the 70s, the BCR-ABL mutation and the corresponding hyperexpressed tyrosine kinase has been identified. In 1992, researchers have conducted HT8s, which stands for high throughput screening, and structure-based drug design studies to identify a class of 2-phenylamminopyrimidine molecules which acted as antagonists of the hyperexpressed protein. Later on, 4 years later, in 1996, this class was optimized by addition of methyl and benzamide groups to yield imatinib. In 1998, the clinical studies started, and in 2001, FDA approved imatinib for the treatment of chronic myeloid leukemia. And in 2016, due to its impact in cancer treatment worldwide, imatinib was added to the WHO Essential Medicines list. Another example of drugs developed by the rational drug design methodology are the HIV protease inhibitors. So in the early 80s, HIV was identified and was linked as the main cause to the AIDS syndrome. In 1985, the HIV protease enzyme was discovered, and its role in viral replication was identified. Later on, with the advancement of X-ray crystallography, the crystal structure of the HIV protease was resolved, and in 1994, sakinavir was approved by the FDA after a structure-based drug design approach. In later years, 2 years later, and between 1995 and 1996, further optimization led to the approval of ritonavir and indinavir. And in 2006, with the later optimization, we discovered second-generation protease inhibitors such as dazabuvir. This highlights the importance of rational drug design and especially structure-based drug design. Since 2 years after the discovery of sakinavir, the annual death rate of AIDS patients dropped by 60% in the US alone. Now, to move on to drug repurposing. So the main problem with the classical drug discovery scheme is that we have only a handful of molecules that reach phase 3 clinical trials. It has been estimated that between 1997 and 2007, only 400 molecules per year reach phase 3 clinical trials, and only half of them are usually approved and later on commercialized. So to resolve this dilemma, and as many drugs exhibit polypharmacology properties, and that they simultaneously act on multiple targets or disease pathways, regardless of whether they were targeted to a specific molecule. Drug repurposing or repositioning aims to take a drug which can be approved or in advanced clinical stages, or even a drug that has been withdrawn from the market, thus a molecule that has undergone extensive safety and efficacy testing, and use it for an additional or an unrelated indication. Drug repurposing comes with numerous advantages, such as lower costs, estimated at less than $500 million per drug repurposed, shorter timelines, with the main time of clinical use put between 3 to 12 years, and of course leveraging existing safety data. It also comes with fewer clinical trial and preclinical requirements. Repurposed drugs may also have well-established dosing regimens and formulations which can further expedite their entry into the market. So, about the methodology of drug repurposing. For a classical drug, which you have on your left of the screen, we usually have an action on a target and an indication and potential side effects. Drug repurposing can be made by 3 main approaches, either drug-centric, target-centric, or disease-centric. For drug-centric repurposing, we expand the application of an existing drug to a new target and thus a new indication. For a disease-centered drug repurposing, we match a disease with no treatment or with an ineffective treatment with drugs that can exhibit better therapeutic impact. This can be accomplished using computational models that detect similarities between features of diseases and predict that similar drugs can be used. And this approach is particularly useful for rare diseases. Then finally, we have the target-centric approach, which matches a new indication without a treatment with an established drug and its known target. The old and the new indication typically differ significantly. And of course, all previously described rational drug design methods, such as docking, QSAR, virtual screening, as well as clinical, pharmacological, and physiopathological data can be used for drug repurposing. The opportunities and challenges that come with drug repurposing are numerous. For the opportunities, we have accelerated development timelines, existing safety data, cost efficiency, rare and neglected diseases, and of course the combination of therapy. For the challenges, we have the limited funding for repurposing research. Drug repurposing may not attract as much funding as the development of new drugs, especially from private investors. The lack of incentive for pharmaceutical companies whom may be hesitant to invest in repurposing research due to limited opportunities for patent protection and market exclusivity. Identifying new indication, which requires a comprehensive knowledge of the drug's pharmacology and disease mechanisms. Regulatory challenges, since repurposing may involve using drugs for conditions outside their original approval, leading to additional requirements and uncertainties. And finally, clinical trials complexity, especially when the drug has a well-established safety profile, and determining appropriate dosages and patient populations can be complex. Now, to move on to some examples of successful drugs that have been repurposed, we can look at aspirin, dexamethasone, thalidomide, and epsilon. So aspirin was originally marketed as an analgesic drug in 1899. It was later repurposed as an antiplatelet drug, and then in the late 80s, leading to its widespread use for preventing cardiovascular events. Ongoing research suggests a potential new repositioning of aspirin in oncology, and it has been shown to reduce the risk of developing certain cancers, such as the colorectal cancer. For dexamethasone, initially, it's used for various inflammatory and immunosuppressive conditions, including asthma and rheumatoid arthritis. Dexamethasone is a corticosteroid that was evaluated in clinical trials during the COVID-19 pandemic, reducing mortality in hospitalized patients requiring respiratory supports by dampening or lessening the immune system's overreaction to the virus. Then for thalidomide, which is known for its tragic history and its banishment by the WHO in 1962 due to its harmful effects on developing fetuses. It was first repositioned in 1998 to treat an autoimmune complication of leprosy, and then in 2006 it was repositioned another time as a first-line treatment for multiple myeloma. Finally, we can look at epsilon, which is a seleno-organic drug that has been used in the treatment of noise-induced hearing loss and bipolar mood disorders. And recent studies have shown promising antiviral activity against the SARS-CoV-2, and it's currently undergoing phase 2 clinical trials for COVID-19 treatment. So to conclude this presentation, we can agree that both drugs design and repurposing are critical areas in modern pharmaceutical sciences. Drug design leverages innovative methodologies like rational drug design, high throughput screening, and computational tools to create targeted therapies. Meanwhile, drug repurposing offers a time and cost-effective alternative by identifying new applications for existing drugs, significantly accelerating the path to clinical use. Both approaches face challenges such as high costs, lengthy timelines, and scientific uncertainties. However, advancements in artificial intelligence and structural biology and big data are transforming these fields, promising more efficient drug development and broader therapeutic applications. And thank you for your time.
Thank you, Dr. Madeleine,
Thank you.
for this great talk. So the stage now is open for questions. If you have questions, please raise your hands or anything else on the table. Initiate discussion. So I have a question for Dr.
Yes.
Madeleine. For a candidate drug that is repurposed, do we need to have the clinical trials from the beginning for an application?
Yes, so for another application, so it depends on the mode of repurposing. If we repurpose indication or formulation, either way, it will be submitted to a regulatory authority as a variation. For new indication, we will only require phase 2 and phase 3 clinical trials. But for new formulations, which is also known as a drug reprofiling, it will be a whole other submission. So we will need the whole dossier going again through phase 1, phase 2, and phase 3.
What is the difference between drug reprofiling and repurposing?
So the definition of drug repurposing is not really clear, but drug reprofiling mainly refers to changement in the structure or in the pharmaceutical formulation. So adding an amine or a methyl group would be reprofiling, and also changing a local drug to a systemic drug will also be considered as reprofiling.
Yes.
And I guess the question concerning IP. So every drug that's now at the market, where a big company has the IP, definitely cannot be repurposed, right?
Yes.
Just by the company itself.
Yes.
So when you go to old drugs, which company would be interested in selling this drug? Of course, you don't get IP again, so every company can sell it.
Yes. Yeah. So for IP, for intellectual property, that's what makes basically drug repurposing a bit challenging, because the legal background is still unclear and uncertain. However, from what I found, FDA grants a 3-year IP protection for repurposed drugs.
Is this sufficient to cover for the costs? I mean, just when you have a patent, if you just bring your drug to the market, you have maybe 5 years.
Yes, but the costs are significantly lower. We talk about $500 million compared to more than $3 billion. So I guess if some companies envisage drug repositioning, that it's profitable in a way. And of course, a lot of drug companies are now selling their old, well-established drugs, since most of the pharmaceutical industry is now turning to innovative drugs, and these well-established drugs can be subject to further drug repurposing.
One question? Okay. What is the main difference between structure-based and ligand-based? You have said that structure-based improves the identification of binding site. So for that, don't we need a ligand to identify the binding site?
No, the difference between them is simply whether or not we know the structure, the 3-dimensional structure of the target. If we know it, it's structure-based drug design. And if it's unknown, it's ligand-based drug design.
One question? We can move to our
Okay.
next speaker.
Thank you.
So it is my pleasure to introduce Dr. Kaiser Dira. So he is a researcher at Pasteur Institute, holding a PhD at this institute and HDR in bioinformatics, with large experience in bioinformatics education and training, and the supervision of multi-omics and bioinformatics projects. So Dr. Kaiser is the head of Laboratory of Bioinformatics, Biomathematics, and Biostatistics in Pasteur Institute since January 2024. He is also PI and co-PI and work package leader for several national and international projects, including the Afrigene D, Find Access, Triomix Curve, MetaCurve, and other several projects. He initiated the first multi-institutional research master in bioinformatics in Tunisia and was highly involved in building capacities in bioinformatics at several institutes. He also led the implementation of HPC cluster in IPT and assisted researchers in order to run their bioinformatics job. So Dr. Kais is author and co-author of more than 60 publications in highly impacted journals in bioinformatics field, and he is also co-editor of 4 Intake open books. So Dr. Kais, the stage is yours.
Thanks, Nasreen, for this kind presentation, introduction. First of all, before starting, I would like to thank all the organizer, and especially Dr. Lilia and also Dr. Nasreen for this invitation. So it is a great pleasure for me to to be part of this event. So thank you again. So my presentation today is titled Advances in Genomics for Drug Repurposing. And through this presentation, I will talk about drug design or drug discovery versus drug repurposing, but I will focus mainly on in introducing some bioinformatics approach using for drug repurposing, and I will give an overview of what we are doing in our lab in the frame of drug repurposing for some diseases. So I will start by a brief history of drugs for rare diseases. So the Food and Drug Administration, FDA, has approved more than 500 orphan products and rare disease therapies, and currently make up more than 50% of new drug approvals of FDA. And this is a beacon of hope for the over 25 million Americans living with one or more of the roughly 7,000 known rare diseases. So the modern process of discovery and development for a drug includes, and it has been showed by my colleague Dr. Radwan. So it includes target identification, high throughput screening of small molecule libraries for hit identification, investigational new drug filing, clinical trials, filing for final FDA approval. And once an investigational new drug is approved by the FDA, so it needs many other steps, including phase 1 clinical trial of the experimental drug that will be initiated. Then phase 2 and 3 trials need to be conducted before the drug is approved and goes to the market. So as you can see here, and as you may know, so the drug development process here, so for the de novo drug discovery, it took from 10 to 17 years and comprises basic discovery, drug design, in vitro and in vivo experimentation, clinical trials, and finally drug registration into the market. So the process is very long. And also the process is highly costing. Okay, so initially, as you can see, so we start by 10,000 compounds in the beginning to end up with one molecule that goes to the market. Okay, and concerning budget, as you can see here, so the cumulative cost per one drug can equal $3 billion. So the process of drug discovery and development is very lengthy and very costly. So as an alternative to drug development or drug design, so we are talking about drug repurposing. So what is drug repurposing? So drug repurposing or drug repositioning, or also we talk about reprofiling or retasking, is a strategy for identifying new uses for approved or investigational drugs beyond their original medical indication. Okay, so this approach leverages the known safety profiles and pharmacokinetics of existing drugs and will speed up the process of making new treatments available to patients. Okay. So the drug repurposing pipeline in oncology, but also in other diseases, began to gain importance in 2000s, leveraging omics data and artificial intelligence technologies. So as examples and of drug repurposing, and the most known, so we talk about aspirin, which initially developed in 1899 as analgesic, but later on used it as antiplatelet aggregation drug. But also we have the thalidomide. Okay.
Okay, it's no problem. ليش قاعد تبدل البيت؟ تبدل هي تبيعات بيبيت
Okay, so sorry about this interruption. So the second example is thalidomide, developed in 1962 and was repositioned as an orphan drug for complication of leprosy. So, and in 2006, the FDA approved the use of thalidomide as a first-line treatment for multiple myeloma due to its anti-angiogenic activity. So this is the new indication of thalidomide. So here, a table that shows the difference between drug discovery and drug repurposing. And as you can see, for time consumption for traditional drug discovery, it can takes 10 to 15 years or more. But using the drug repurposing approach, this time is shortened and can take 3 to 9 years. Okay. Regarding the investment costs, so for drug discovery, it's highly costs highly, but it can exceed $2.6 billion. But using drug repurposing as an alternative, this corresponds to less investment, and the risk of failure for drug discovery, about 90% of drugs fail during clinical trials, but using drug repurposing, so we can lower this risk of failure. And regarding the clinical efficacy and safety evaluation, so for drug repurposing, this already exists and often supported by previous use in different indications. So what are the different steps and the workflow of computational drug repurposing? And here we are talking about bioinformatics. So there are mainly 4 steps. So the first step is the strategy selection. So the first step is to decide on the drug repositioning strategies. So what kind of data we will use, so genome, phenome, or a combination of 2 or all of them, etc. So this is the first step to perform. The second step is the approach selection, because here we have many approaches that can be used for drug repurposing, and we need to decide on the computational drug repurposing approach. We can use data mining, machine learning, network analysis, or also the combination of many approaches. The third step is the model validation. So we need to validate models by using evaluation metrics, cross-validation, accuracy, etc. And the fourth step is the prediction and the application. And here we need to predict therapeutic potentials and novel indication for existing drug, and how to utilize model in diseases areas such as cancer, rare diseases, and so on. So as was nicely stated by my colleague, Dr. Radwan, so we have 3 types of approaches that can be used here. So disease-centric approach, target-centric approach, and drug-centric approach. And for the first one, we begin with the focus on a particular disease. The process includes develop disease model to understand underlying biology and key pathways, and we need to utilize pathway analysis, network biology, and system biology approaches to uncover new mechanisms of action. For the second type or second approach, so is the target-centric approach. And here, the emphasis is we will concentrate on a specific molecular target or pathway, and we will employ computational methods like machine learning, graph neural networks to predict drug-target interaction. And we can use databases of approved drugs database screening to identify potential therapeutic for new indication. And the last approach is drug-centric. So the focus is to find new use for existing drug And the method includes screen compound libraries to match approved drugs or clinical candidates with novel biological activities. So here we employ high throughput screening to test compounds. So we perform screening against various biological assays. So here, an overview of the different approaches. So bioinformatic approach, we can use, as I say, data mining, network analysis, transcriptional signature, protein comparison, binding site detection, docking, ligand similarity, etc. So now I will talk about small molecule drug development for rare diseases, and I will focus on the signature-based approach. So what is what we mean by this approach? So this is a method that explores existing drugs for new therapeutic application by analyzing their molecular signature based on transcriptomic data, okay, in relation to specific disease signature, okay. So rather than targeting a single molecular pathway, this approach looks at the broader interaction between drugs and diseases, seeking potential matches based on shared molecular characteristics. So this strategy includes several steps. So the first one is to gather transcriptomic data from diverse sources like NCBI, European Bioinformatics Institute databases, or DDBG, and generate drug transcriptomic profiles using high throughput techniques, so that you can find a different database. The second step is the data integration. So we will integrate transcriptomic data with other omics data and use advanced data mining to identify some patterns. The third one corresponds to the generation of the signature that define unique molecular signature for diseases or drug responses. The fourth step is to filter and select drugs with similar signature and structure. And the last step is, of course, the validation of selected drug for repurposing potential confirmation. So here, an example of the signature-based approach for lung cancer. So for, as you can see here, so we start by transcriptional signature of lung cancer. So we have different type of lung cancer, so small cell lung cancer or non-small cell lung cancer. And for each type of disease, we have what we call the connectivity map. So for each disease, we have the signature, transcriptomic signature related to each disease. And in addition to, so this kind of data you can find in different databases like CMAP or links data, okay. And for your case, just you have to integrate your transcriptomic data related to one patient for one disease, okay. So, and compare the patient signature with what we have in the different database, and try to correlate your signature with one drug. Okay, this is the main approach. So the advantages of drug repurposing in general. So as I say, to reduce the cost, you can combine therapy. It's the nice way for personalized medicine. Faster the development timeline, increase opportunity to market shelf the drug, etc. So what are the challenges? Okay, so drug repurposing have many opportunities, but also have some challenges. And among the challenges, so we have the clinical trial expenses. Repurposing benefits from completed clinical development stages. However, skipping preclinical study poses some risk. The second point is incorrect drug identification may lead to minimal therapy. The third point is the therapeutic indication selection. So choosing suitable therapeutics area poses significant challenges. The regulatory requirement and we talked about. So inadequate data may necessitate new preclinical and clinical trials to meet regulatory standards. We have also the problem of intellectual property, so and this poses hurdles for repurposed drugs, and also the market demand. Increasing demand can incentivize researchers and investors to engage in repurposing efforts. So here are some example for in oncology. So you can find many therapies in blue. I will give some examples. So you can see that aspirin here is the example of aspirin, also the PARP inhibitors, the doxycycline, the statin, etc. So there are many drugs that have been repurposed mainly for treating cancer, but not only cancer, so it can be applied to other diseases. So now I will give one example of what we are doing in the frame of our lab and in the frame of Ghada and Rehab masters. So we have developed a pipeline for drug repurposing in oncology, and as I said, so here we used the signature-based approach. So we extracted all the data that are available in CMAP and links data, and we have identified or developed a pipeline, an automated pipeline development using reference database, and make the use of this web interface interactive for user. So here is the whole process. So we started by data acquisition, as I said, so from different databases when we can find signature related to different diseases. So data have been reprocessed to identify differential expression for each kind of disease. We performed the gene annotation. We identified for each gene signature, we performed drug analysis, and we calculated for each disease a score. Then the result is visualized, so in the frame of tables or heat map, etc. And this, as I said, is based on gene expression data. So the key steps were the differential gene expression analysis, gene annotation, drug repurposing via CMAP small, but also links data that have been integrated. And so we were able to identify some drugs with significant negative scores that are likely to reverse the cancer-related gene expression patterns. So here, just a screenshot of the web interface that has been developed and is under improvement. So we have developed a front end, so user-friendly interactive design. So we have also the backend streamlined workflow for dataset upload, bioinformatic analysis, and result visualization. And the purpose is to simplify complex drug repurposing signature-based approach workflow for researchers. So as I said, so this is this work was performed in the frame of Ghada and Rehab masters. I will finish by this slide. So while drug repurposing has shown immense promise in oncology, its potential extends to various other fields, can include neurology, infectious diseases, rare diseases, cardiovascular diseases, autoimmune diseases, respiratory diseases, and so on. So it can be applied to all kinds of diseases that you are working on. So I would like to acknowledge my master students, so Ghada Rehab, Usama. So Ghada Rehab are both master student of myself and Usama, the laboratory, the Institute Pasteur of Tunis, and here I would like to acknowledge all the team. Thank you very much.
Thank you. Is there any question?
What is the difference between Ghada and Rehab?
Thank you, Faisal, for your insightful presentation.
Yeah.
What kind of data input your platform take? So show you how.
Yeah. So just as input, they take just I will show you. So it can take sorry here. Yeah. Just it can take expression data, so CSV file of analyzed transcriptomic data, or it can directly take gene set, so GSE accession number from gene expression omnibus or array express. Okay, so you can directly download from gene expression omnibus data, and it will be processed. All the analysis will be performed here. Okay, so the expression data will be or you can use already analyzed transcriptomic data. So you provide only the CSV file, and it can be integrated in the interface, and you can finalize the process.
And how many samples did you analyze to have reliable results? Because I have the same idea.
Yeah. So currently it takes all the GSU. So you can have 100 samples in your experiment, or just few samples. So it doesn't matter. Okay. So it takes all the file. Also the CSV file, you can have many sample within your CSV, and it can be integrated. Okay, so we are in the process of working on the interface, because it's very basic, but it works. So hopefully we will make this public to all users to use it.
Sure.
Thank you.
And hopefully it will be published also.
Thank you, Dr. Abdira, for this amazing presentation. In fact, I have two basic questions.
Yeah.
I wanted to ask about the percentage of the compatibility between the transcriptomic signature with other transcriptomic profiles. So we say that this drug could be possible for this patient or for this disease.
Yeah.
And the second one, if we can test the combination, you said that we can combine 2 or 3 drugs together. So if we can in silico see if there's no side effects of the combination of these 2 drugs.
Okay, for the first question, so what is calculated here is what is known as a connectivity score. Okay, so based on a signature profile, the tool will calculate a score.
Yes.
If the score is very low, it means that the drugs can fit well with this kind of transcriptomic data. So as a result, we can have let's say 5, or we can also have the top 10 drugs that can fit your but you need to go to further investigation to identify the best one, because
But
yeah.
we have a limit of the score, so to select or not.
No.
Okay.
Okay, so
We just have the best
yeah, exact.
compatible drug.
Exact.
Okay.
So this is for the first question. The second question regarding the combination. So honestly, I didn't. So we are exploring the different approaches that can be used. So currently, so as I said, there are many different approaches. So signature-based, ligand-based, structure-based, etc. So we will finalize this, but we will also integrate the different approaches, and we will combine all the approaches. Okay, so we will try to investigate signature approach, but also we'll work separately. Then we will try to combine in order to give more confidence to our result. So if we find same drugs identified by different approaches, it means that it will be more reliable, let's say. Okay, so this is will be done. So yeah.
Just the synergy between the drug.
Yeah.
Validate from experimental. Like after
Only
in Silico,
experimental.
yeah,
Okay.
it's very difficult to validate.
Yeah, exactly.
Yeah,
You can.
Silico study.
Yeah.
But did you actually think about using your tool against other studies that already identify drugs for repurposing using signature-based strategies? And did you come up with the same molecules?
Yeah.
I mean, this would be very cool. To just go into transcriptomics data of a different lab that use that to get a new drug or to repurpose drug, and you find the same drug
If we have
based on your tool. I mean, this is kind of the best validation you
Yeah, of course.
for.
Yeah, so we will try to test that. So in order to make stool very web interface in the analysis very reliable, so we will probably do that. Yes.
But if we have a problem in the experiment of some problem in the drug,
So we can take several diseases for which we already have we know the drug and we can test and we can see if we find the same result. Yeah.
Another question.
Yeah.
What kind of cancer did you target in your analysis? One cancer or?
I think that the example here is so we tried five types, I think. So breast, colorectal, and so correct me if I'm I'm wrong. So five type of cancer. So breast, colorectal, lung, and two other type of cancer. So for this example, but we will extend. So because it's not limited to one disease, so we can just so links correspond to, so include all the kind of diseases that exist and for which we have already transcriptomic data and associated drugs. So we can use it for
the liquid cancer.
Yeah.
Maybe not a good question. Would it be possible to associate another database to it that has compounds in that are not like FDA approved or otherwise in the future that could be screened against? It would not be repurposing more discovery.
Yes. So now we are only based on signature-based approach, which means that we are using only transcriptomic data, and so. But if we want to extend to use structure-based approach or something like this, so we can of course integrate other databases like drug bank, like other database related to molecules, related to drugs, etc., and we can perform the automatic screening, etc., and we can have another interface, maybe so the same website but another interface, and we can also combine the different approaches together in order. So yeah, we can integrate other database. Yes.
Another question, because it's very interesting. So the molecule that you identified through this pipeline, do you plan to contact pharmaceutical industry for clinical trials, or it's just an in silico work? And
so of course this will be the second steps of all this, but the main steps here is to have this work running correctly and smoothly and etc. So this is our first objective is that, so to make all the process automatic and control all the different steps to avoid any troubleshooting, any bug, etc. So because the process is hard, especially because the databases are very huge. Okay, so links data takes many time to be loaded, etc. So we are finalizing all the steps for this. Of course, after that we will look on the biological side of all the, so to be sure that our results are very reliable, and of course we can take the different more interesting molecules and go further.
Good luck for that.
Thank you.
One question
Thank
from
me.
Hussein.
Yeah,
It's a very technical question, right?
technical. Okay.
Yeah. So last time I checked the drug bank database.
Yeah.
The data that they were put that it was possible to download it via an XML file and
Mhm.
then do the parsing of this file.
Mhm.
So, but I think that now this step has changed. So, how do you integrate the database?
You are talking about the drug bank, right?
Yeah.
So yeah, here for signature based, we are not using drug banks. So we are using links data, which are free, and no problem for that. But if you plan to go to structure based approach, of course we need the drug bank, and it will be maybe it will be a problem for us, but I don't know. But so for drug bank we need to contact them to get the data, because it's not open. So I already did it 2 to 3 years ago, but of course I think that they have changes in their usage or something like that. So we need to contact them again. And of course, I think so. They made a lot of updates in their database, so we need to recontact them in order to get the new version of the data. Yeah.
Other questions? So thank you for this.
Okay.
Are you only planning to limit this project on the molecules for cancer, or are you going to try molecules for neurodegenerative diseases, as they are considered as rare diseases, but the prevalence now is increasing, and in Tunisia we have aggressive.
Yeah, I got your question. So thank you for this question. So just I will put this work in his context. So just it is work in the frame of a master. So we need to focus on a kind of disease. So we focus on oncology, but this work can be applied to any other disease. Okay, so the data, the links data is not specific, or CMAP is not limited to cancer. It englobes cardiovascular disease, neurology, etc. So if you have your data set related to neurology, you can put it here. You will have drugs that are relevant to your disease. Okay, so it's not specific to, but in order to validate the master, we need to have
With our president.
time. Yeah, exactly. Okay, so yeah. Thank you.
Okay. One more question.
I'm just wondering if this pipeline will be an open source pipeline, like the next pipeline.
Open source. So the interface will be public, but the code will not be public. Okay, so the interface hopefully will be public. So the aim of this work is to make all people doing so, trying this and doing this automatically. Okay, so rather than do it by command line, so you will have an interface for that. Okay.
More details on the paper. We cannot produce the
Yeah, of course.
Okay.
Okay.
Thank you, Dr.
Thank you very much.
Nice start. So now we have to move to
Okay, can you hear me? So I may start. Okay, thank you. So good morning. First of all, I would like to thank the organizer of the workshop and specifically Dr. Ramdan for her kind invitation. So as she previously mentioned, we are currently struggling to travel, which was the reason why I wasn't able to make it in person with you. So therefore, I'm going to do my presentation online, and let me know if you have any questions later on at the end. So the title of my presentation is Computational Drug Design with an emphasis on the role of structured bioinformatics in the drug design project. So I will start with this outline. So I will give a brief overview on computational drug design with the role of structured bioinformatics. I will then compare a little bit the use of experimental versus predicted structures. And then I will move to the interaction analysis. This is exactly where structured bioinformatics could be of a great use for the drug design project, specifically to understand the interactions and optimize certain compounds. And then from molecular dynamic simulation, I will talk on a few slides about the role of molecular dynamic simulations. And I will finish my talk with a few case studies and then the challenges and future directions. So computational drug design, as you may imagine, is the use of computational tools and techniques to discover, design, and optimize drug candidates. And this is by predicting their interactions with a certain biological targets. So as my previous, as the previous speakers mentioned, that in a traditional drug design project, you could encounter many challenges related to the time, the cost, and the high failure rates. So as previously mentioned, that usually a drug design project, it's a very lengthy, time-consuming project that could cost over $2.5 billion on average, with a very high failure rate, especially in the late stages of the project. So computational methods can significantly cut down the drug discovery timelines. It can reduce the cost with, in fact, and it can as well improve the success rate. So what is the role of structured bioinformatics in the drug design project? In fact, structured bioinformatics will play a very important role in understanding the existing interactions that you could have between a certain drug and its target. And this could be explored, as I mentioned, in order to optimize and improve certain drugs by exploring, in fact, the binding site of a certain target in order to make it, or in order to improve the affinity of the drug. And as well, later on, I will explain that you can even use structured bioinformatics approaches in order to reduce certain, or to make the drug more selective to a particular target. So what is, again, structured bioinformatics? So structured bioinformatics, it's a branch of bioinformatics that studies and predicts the three-dimensional structure and function of biological macromolecules. So this is a growing research area, especially when you compare it to experimental procedures that are inherently low throughput and very expensive to achieve. So I can give you a very small number when it comes to determining an experimental structure versus predicting its structure. So in fact, determining experimentally using, for example, X-ray method, it costs approximately $350,000 in average. While in fact now anyone can predict a structure using, in fact, those AlphaFold approaches that are very cheap in terms of cost, because you can run everything on your computer. So this is what makes now, the computational approaches, and more specifically, structured bioinformatics is a very interesting, area to discover. So in structured bioinformatics, you could have, again, as we said, you can understand the interaction that happened between a certain drug and its target. And therefore, you can understand certain mechanism as well of binding, of inhibition. And based on that, you can study as well, for example, mutations that could lead to a resistance. So all this, in fact, could be achieved, using structural data and more specifically, those computational tools. So one of the most, again, impactful application of structured bioinformatics is in drug discovery, which is mainly, the topic of this workshop, because by understanding the three-dimensional structure of a target that is, or target protein that is involved in a certain disease, a researcher can design molecules that precisely fit into the protein's binding site and inhibit its function. So let me show you the different contributions that you could have when it comes to structural bioinformatics. So first, as we mentioned, you can reveal structural details. So in this case, you can identify active sites, binding pockets, structure motifs. You can predict the interaction that you could happen or that could exist between a drug and a certain target. And of course, you can go into the comparative studies. In this case, you can analyze the evolution of, or the conservation, in fact, during evolution of structures. And therefore, this could help as well to predict the function by looking at structural similarities between, in fact, a protein with an unknown function and other proteins that are very well characterized. So when it comes to the databases, of course, there is a long list of databases that we use in structural bioinformatics. Having protein data bank being typically at the top, because this is the main source of structural information for the different biological macromolecules that we have. And of course, recently with AlphaFold and typically all these predicted models that are being stored now in AlphaFold DB, or the database for AlphaFold. And of course, we should not forget the UniProt, which is in fact a very useful database for sequence annotations of proteins and other information related to proteins that we could profit as well in our drug design project, and more specifically, in fact, in the computational drug design project. So when it comes to tools, we do have a lot of tools. So I'm just going through the main important tools that we use typically. So we do have tools related to visualization and analysis. So these are the 3D visualization tools that we use in order to visualize our results, the interactions. So we do have Chimera, we do have PyMo, we do have VMD, and other, let's say, online plugging that we can use as well to visualize, in fact, our protein complexes. So whenever the experimental structure is not available, in this case, we can predict, the structure of our mainly protein, and definitely in the new AlphaFold, AlphaFold 3, we can predict as well now the structures of other biological macromolecules going from DNA, RNA, so mainly RNA, let's put it this way. So in this case, AlphaFold is the most famous between these structure predictions these days, followed by Rosetta, by iTaser. You can go for Modeler as well for comparative modeling. So all these, in fact, are tools that you could use for your structure prediction. And when it comes to docking, so again, I will be talking more about dockings in detail later on. So docking, you have, again, a list of softwares that you could use. Some of them are free for academics. Some of them, unfortunately, are commercial, and you have to buy a license. So, and finally, once you're done with the docking, this is where you come for molecular dynamic simulations. So the most famous free for academic software is typically GROMACS and SHARM. The others, for example, Ember, you have to buy, a license for Ember. But definitely you can do a lot only with, GROMACS and SHARM. And then all this, approaches, so whether starting from basic visualization to simulating the motion of or the dynamics of how a protein interact with a ligand, all this, in fact, is very important to provide data. And this data, again, it has to, again, you cannot consider this results to be, let's say a confirmed results. That has to be, in this case, or that could serve to guide the experimental drug design, or even be validated by some experiments. And therefore, you can go back to your computational approach and improve those in order to improve your predictions, or even to improve, the different outcome that you can get from these softwares. So if we consider a little bit the experimental structures, so I updated, these numbers a few days ago. So we could be slightly in the 228,000 right now. So the total number of structures in the protein data bank is typically now in the almost 228,000 different structures. These are divided into 86.3% that goes into the X-ray, 7% in the NMR, and 6.4% in the electron microscopy. And of course, you may have, a fraction of percent left, and this is typically the other programs. So even if we are dealing with experimental structures, these structures in fact are not perfect, or they cannot directly be used for drug design projects, especially structure-based drug design project. So the challenges that we could have first is the availability. So we have many interesting targets that could be involved in many diseases, but unfortunately, an experimental structure is not available. So it's very difficult to obtain some of the structures for few proteins. And as I mentioned, previously, the process or the experiments are very expensive and time-consuming. And whenever you're looking to use some of these structures for docking, for example, or for structure-based drug design, you will be looking for high-quality structures. And this is to ensure, in fact, the accurate modeling of the interactions between a ligand and a target. So this is where my quality checklist, is typically, designed for this purpose. So we will be looking for different criteria about a certain structure in order to see whether this will be suitable for, in fact, structure-based drug design, yes or no. So first, we do have the resolution. Of course, resolution is typically applied to X-ray and electron microscopy. In fact, we are looking for a resolution that is less or equal than 2.5 angstrom. Keep in mind that the resolution is typically the parameter that refers to the smallest separation between planes of the atoms that are diffracting your X-rays. So the smallest the value of your resolution, the better the quality of your structure is. And we are looking for a resolution that is less than 2.5 angstrom, because this is where you can see the correct positioning of the side chains, the correct positioning of certain atoms, of course, except the hydrogens. And this is very important in order to have a clear identification of the binding site and the different, features that you have in the binding site, like the hydrogen bonds, acceptors, and donors, for example. The other criteria that is very important is the completeness of the structure, because unfortunately, we do have many structures that are deposited in the PDB that are incomplete. Of course, if this part that is incomplete is far away from the binding site, in fact, we can proceed with certain computational tools, but we have to make sure that there is no missing residues in the binding site so that we can, proceed with, the structure-based drug design. Depending on the system that you're studying, you could have sometimes cofactors that are needed, or even ions or water that could exist, in fact, in the binding site. So in this case, you have to include any biologically relevant elements in your docking, for example, calculations. So many systems, for example, they require a cofactor in order to do, the enzymatic activity, or sometimes they need a certain ion, etc. So all this, in fact, needs to be considered whenever you're choosing your target, whenever you're choosing, let's say, the experimental structure for your target. Another, criteria that is very important, it's related to the protonation states. So most of the time, especially with the PDB structure that we have in the protein data bank, the resolution, in fact, doesn't allow us to see the hydrogen bonds or the hydrogen atoms, except for definitely if you are dealing with an NMR structure. But as you can see, the majority of the structures that we have deposited in the PDB are determined by X-ray and by electron microscopy. And this will not allow us to observe, or the hydrogen atoms are not visible. So in this case, you have to be careful on the protonation states. So when you are adding your hydrogens or the missing hydrogens to your structures, you have to take into account the protonation state, the physiological pH of your system, because I can give you a very simple example of a histidine, for example. So a histidine could exist in 3 different states. 2 of them are, neutral, and 1 of them is protonated. So depending on the environment of the histidine, right, it could be in one of those 2 neutral, and it could be as well positively charged. So all this could impact how a ligand could bind to the binding site by impacting, in fact, the hydrogen bonds that could occur between, in fact, the ligand and the target. All this, in fact, are very crucial for the accuracy of the results, coming from the docking. Binding site validation. Most of, for example, the structure-based approaches, they require that we have a prior knowledge about about the location of the binding site. So in this case, we can use what we call co-crystallized ligand. So in this case, the co-crystallized ligands could give us an idea on where the binding site is, but definitely this is not as simple as it is, because we could use, for example, an allostatic binding site in order to inhibit, our protein, or sometimes depending on the drug, depending on the binding mechanism of the drug, this could induce what we call an induced fit that is different in the binding site between the different drugs that we could be testing. But definitely, being able to identify or have a first idea on the location of this binding site is something that could be a good step in order to know where the docking should be, and how we can improve, in fact, the accuracy in predicting the different poses. So I will be discussing more docking in a few slides. And finally, the biological context. So again, whenever we're going to start with any drug design project, we're going to collect as much information as we should on the target, on the disease that we are trying to inhibit or to cure. In this case, make sure that the conformation that you are choosing for your docking studies, for example, or for the structure-based drug design in general, it does reflect, let's say, the biological context of this particular target. So, because definitely, if you're choosing the active state of an enzyme, this could give you, in fact, a more accurate results than, for example, an inactive state that could be very misleading, in the docking results. So when it comes to predicted structures, so as I mentioned before, this is a very important computational aspect that we could have, especially when a good or when just an experimental structure are unavailable. So I'm showing you on this table the different category of methods used for protein structure predictions in general. So it goes from comparative modeling to threading to ab initio, and finally, of course, within the last few years, we have been witnessing more and more the use of AI based approaches in different aspects, coming from predictions, coming from let's say, as mentioned before, machine learning, data mining, in order to come up with predicting certain activities using those type of approaches, and not directly using a docking approach. So AI has been used a lot lately. So again, for the sake of time, I'm not going to go much into this table. So each one of these approaches will give me a certain advantage over the others. So many people now are going directly to the AI-based approach because they think that this could be more accurate. But in reality, it depends on what you have as type of information. Because if you do have a good template for your protein of interest, for example, a comparative modeling could outperform the other approaches. And this is, for example, what is happening a lot in different type of proteases when you're trying to do drug design project on proteases. So if your protease of interest doesn't have any experimental structure, a comparative modeling using certain existing templates coming from different species or other type of proteases, this could be more than enough to get a good quality model that is suitable for the docking, especially that this comes, in fact, at a faster speed than those more advanced approaches like threading, ab initio, or even the AI-based approach. So I'm listing a little bit for you here some of the tools that are very famous in every category. And of course, with an emphasis on the AI-based, especially, AlphaFold. So again, they do have, the advantages of these approaches, that they could give a very high accurate, or they could have, provide you in models with very, accurate, details, especially when there is no homologs that is available. And those approaches are very excellent for large-scale prediction. But keep in mind that these approaches, especially when it comes to protein structure prediction, they are computationally very intensive. So it takes more time than, for example, a comparative modeling. And of course, they do not take into account the dynamic of the system or the dynamic of the protein. Or, for example, if you have a certain disordered region that your protein contain. And definitely in this case, these are usually ignored, or these are not very well predicted when it comes to disordered region. So to move on now to the docking. So a docking approach is, for example, this is a current project that I'm working on. It belongs, in fact, it's a glycosyl transferase enzyme. It could be involved in a certain disease, and we're trying to come up with certain drugs that could inhibit, in fact, this glycosyl transferase. So again, the docking, usually it is the first approach that people will go for when it comes to structure-based drug design. So this is a very fast method to predict the binding pose. So the binding pose, it means the conformation and the orientation of a certain ligand within the binding site of a certain target. And this is to study the affinity and the activity and how this drug interact with your target. So this is, again, a very important, or it plays a very important role in the computer-aided drug design, because this will allow you to screen large libraries of compounds, and I will show you a certain case study later on. And more specifically as well is what we call hit-to-lead optimization. And the hit-to-lead optimization will not be possible if you don't have the structure of your target that you could explore in order to refine certain hit compounds. So again, the hit are usually what we call the initially identified compounds from screening. So the purpose is to try to improve those into lead compounds. So the lead compounds are usually compounds with an improved properties for drug developments. In this case, they have better binding affinities, they have better selectivity, and even, for example, ADMET properties. So all these, in fact, will be typically, especially, when it comes to improving the binding affinities and selectivities, this is something that you can do when you have, in fact, the structure of your target, and you can explore the structure of the target in order, to see where you can modify your ligand in order to improve its affinity. So, but keep in mind that for every docking method, you need to have two components. So the first component is what we call the docking algorithm. So the docking algorithm, this is typically the component that will allow the software to explore the conformational space of a small ligand or a small molecule in the active site of your biomolecules. And the other component is what we call the scoring function. So most of these scoring functions are based on what we call molecular mechanics or force field approaches. And the scoring function is usually what we use in order to predict the affinity of a certain drug to its target through the evaluation of the existing interactions between the two. Okay. But those two, in fact, they will make very few challenges in the docking that I will be discussing, in fact, in the coming slide. So before, you do have different practical issues that you may have to consider when you're doing a docking. So again, even if docking could be seen by some people to be, let's say, the easiest, structure-based computational approach, you do have many practical issues to consider. So one of the previous speakers was mentioning typically the preparation of the input, the preparation of the receptor. All these, in fact, they are very important to do. And in addition to that, you do have different docking approaches. Each of the docking approach is going to treat the ligands or even the receptor in a flexible or a rigid approach. So definitely deciding on whether you will do a rigid docking or a flexible docking, and whether the flexibility is only for the ligand or even for part of your receptor, this is something that is very important to consider. Of course, some of you may directly tell me that, okay, flexible docking is usually more accurate because you are simulating the flexibility of the ligand. But keep in mind, the more flexibility I'm going to take into account, the more computational the cost is going to be. So if you're doing a high throughput screening of large libraries of millions of compounds, you cannot do a flexible docking because it's going to take too long. So this is where people, they go, for example, for different approaches, going from something that is less accurate, but the data is too big to handle. And then they're going to go for approaches that are now more computationally expensive, but will lead to a more accurate results. Okay. So the speed is something, again, this is what I call usually the compromise between speed and accuracy, right? Because of course, everyone would like to have the most accurate result, but the speed and the computational resources, it could be very limiting sometimes, okay? So this is, again, a practical aspect, and the scoring function. So in fact, we do not have a perfect scoring function in docking, right? So we do not have a universal scoring function that will perform very good on all the scenarios, on all the complexes. So again, a perfect scoring function is usually a function that will accurately calculate the binding affinity between a drug and the receptor, and it will score the poses of an active drug higher than the pose of an inactive. In addition to that, it will score the correct pose of the active higher than the incorrect pose. So again, imagine, let's say we have 10 different compounds that we're testing. 2 of these, in fact, they are active on my receptor, 8 they're not. So a good scoring function will allow me to score those 2 actives higher than the remaining inactive. In addition to that, remember, the docking is going to explore the conformational space of the ligand inside the cavity of your receptor. In this case, I should be able to score the correct pose higher than the incorrect pose. Okay, so definitely you have a big, or the results of the docking is highly impacted by the quality of the scoring function, and of course by the quality of the search algorithm that will be able to find the right pose of the ligand inside, the binding site of your receptor. So for this reason, usually we go after, filtering many of the drugs. We go, for example, for molecular dynamic simulations. Why? Because in the molecular dynamic simulation, we simulate what we call time-dependent motions. And we know that the biological activity of any biological system in our living organism or on any, living, species, it is in fact, it's time-dependent, and there is a motion that is going on, there is a dynamic. So in this case, molecular dynamic simulation is going to generate confirmation of your system at different time scale. And this is highly valuable because it will allow me to refine my docking results. But again, you cannot do it on a big list of ligands. You will do it only on few ligands that shows promising results in the initial, docking calculations. So very important to capture the dynamic behavior of protein and ligand complex. It will allow you to evaluate the binding stability if you have any conformational change, and therefore it will allow you to study or to obtain certain mechanistic insights, resistance coming from mutations, or even allosteric sites. So all this, in fact, could allow you to obtain or to be obtained, in fact, from a molecular dynamic simulation. And again, this is more computationally expensive than, the docking calculation. So just to go briefly, so we do have a lot of examples of molecular dynamic simulations in the literature, whether in fact it's for solvated proteins, for protein DNA complex, for lipid systems. So here, for example, I'm showing you, an example of sodium-potassium pump. So again, in order to simulate correctly your system, you need to be as close as possible to the physiological conditions of the system. So in this case, you see our our pump in the ribbon presentations in yellow. This is surrounded by this bilipid membrane, and of course from the inside and from the outside you do have your water molecules, because again it's usually in a solvated or let's say one side of the membrane is going to be an aqueous environment. Same story for the other side, and the purpose in this case is to simulate the motion of how these sodium and pump ions will cross, or at which speed they will cross this pump. And of course in this case you can easily study the impact of a certain mutation, how it could slow down ionic gradient across the membrane. And this is something that we can do only when you start simulating the motion of a certain system. So I know that I could be running off time. So Lydia, you can let me know if I'm exceeding a little bit the time. So just to show you some very, very important case studies. So this is a very good review paper that was published in 2022 about, in fact, the last 15 years of using structure-based virtual screening for drug discovery. And in fact, they do provide you a lot of important, let's say, examples. Of course, about the historical HIV proteases and kinase inhibitors, because this is typically where one of the first examples where they have used structure-based drug discovery. Yes,
Hello?
Lilia. Yes.
You still have 5 minutes. Please try to resume.
Yeah, I'm fine. I'm gonna finish on time in this case. Okay.
Okay,
So in this case, it's a very good review paper that I highly encourage you to read and check, the different examples that they highlighted. So I will choose one very recent, example that was published in JACC about a ultra large virtual screening to identify, in fact, SARS-CoV-2 main protease inhibitors, and the purpose here is to find, in fact, a drug against this, COVID-19 main protease. And the story behind it is, in fact, that drug repurposing did not give you significant results in the clinical trials. And this protein is very important for the cycle of the virus, which makes it a very promising, target of interest. And we had few issues with the previous drugs about, the lack of selectivity and poor pharmacokinetics properties. So in this case, what they did, they went through a structure-based docking where they have screened a library of 235 million compounds against the active site of this, protease. And just to give you an idea, the docking took an entire day on 300,000 different CPU cores. So again, the computational resources needed in order to screen this large library of compounds. And then, of course, they selected the top rank, so the 100 top rank compounds, and this goes into, in fact, binding and enzymatic assays. So they went from the in silico to, in fact, the in vitro and the in vivo. And what they found typically two interesting drugs that they were able later on to obtain the crystal structure of these drugs. So in fact, first they had the docking results. They selected those two compounds of interest, and they went for the experimental to determine, in fact, the crystal structure. And this is just to show you that the docking in this particular case was very successful, with only a root mean square deviations between the predicted confirmation from the docking and the real experimental one that is very small angstrom for compound 1 and 1.38 angstrom for compound 3. But those two compounds, in fact, they were not very interesting in terms of affinity, and they went for the hit to lead optimization. And it was only possible because they had the structure that they were able to obtain. And this is where, in fact, they tried to explore two different cavities in the in this main protease, and they came up with a list of different modifications that they brought to the system or to those new compounds. And what we noticed a clear improvement of the affinities going from a micromolar to typically in the nanomolar range. And this is typically a few drugs now that went into clinical trials. So I will skip the second, let's say, case study. So, and then I would like just to finish on certain challenges and future directions. So despite, in fact, the structure-based drug design to be very successful, it still encounters certain challenges that are related to data quality, model accuracy, and of course, computational limitations. Because if you ask me, I would say it would be better to do molecular dynamic simulations for all the drugs that we're screening. But computationally, in fact, it is not impossible, or it's typically impossible to do. And the future advancement in the field that we have been witnessing for the last, let's say, few years are typically the integration of structural bioinformatics with the machine learning and the AI in general to enhance the predictive power of computational drug design. And even in a recent review that we published, in fact, it was typically to review the use of artificial intelligence in the different steps of drug discovery, and with a focus, in fact, on neurodegenerative diseases. And this paper, in fact, was recently published in Drug Discovery Today. And I would like to finish. Thank you for your attention, and I will be up for your questions. Thank you.
Thank you, Dr. Joseph, for this great presentation. So now, if we have questions, please try to speak out loud or come here closer to PC so Dr. Joseph can hear you. So,
and please, I'm sorry again for all the technical difficulties that I really created for you just because I wasn't able to be in person. Okay.
It's okay. It's okay. Thank you for your dedication and commitment.
Thank you.
Thank you. So, is there any question? Go ahead.
Thank you, sir, for your presentation. Okay. So my question is about docking tools. You've mentioned several tools, but my question is there is special tools for flexible docking and rigid docking. And can you advise us how to choose the right tool?
Okay, so in fact, most of these tools right now, they do both type of docking. So for example, if you consider AutoDock or AutoDock Vinia, for example, or even Dock or Glide or FlexX, all these approaches, in fact, they will allow you to do a rigid docking and a flexible docking depend on your need. So again, the question is, once you decide on the flexible docking, you always have to keep in mind the extra computational cost that is going to create for you. So definitely it's going to be computationally more expensive to explore the conformation space now of the ligand that you are considering flexible, or even if you're going to consider, for example, the flexibility of certain side chains in the binding site. So all these are things to consider. So now if you're going to ask me which, for example, which docking tools is going to be, or I do recommend, it depends on the task. So if you're checking a little bit the literature, you will see, for example, that Dock, and more specifically Dock 3.7, is more suitable for high throughput screening, right, of large libraries. And if you consider, for example, something that is more specific to optimize, for example, a hit to lead or something, in this case, a Glide, AutoDock Vinia, FlexX are usually, in fact, a good docking approaches.
Just to clarify, I think that Amber Tools is free. We can use the.
Oh, the Amber tool is free. Correct. The Amber tool is free, but the engine to simulate the dynamics is not. Yes.
Thank you.
Okay. Any questions? So Dr. Lilia now has a question. Oh, yes.
Hi, Josette.
Hi, Lilia.
Hi to see you. I'm happy to see you. So, unfortunately, you didn't come here. So, but thank you for your commitment and your great presentation. So, I have a simple question. How do you choose the template for docking? Is it any criteria?
So this goes back a little bit to my checklist. Typically, let me put it back for you. So in fact, it's related to the quality of the structure. So first, what I'm gonna be favoring is the resolution, because in fact the resolution is gonna give you the level of details that you need. So the smallest the value of the resolution, the better the quality of your structure is gonna be. So in fact, we usually take a resolution that is less than 2.5 angstrom, because this will give us an accurate positioning of the side chains and a precise positioning of the atoms, again, except the hydrogens, which is in fact needed for the docking. And the second criteria that I will go for it is typically the completeness, to make sure that there is no missing part of your experimental structure that is around the binding site. Because if you have a missing part, let's say for the docking that is very far from the binding site, you could ignore this, it's fine. But what matters is not to have a missing loop, for example, or a missing part that is directly in the binding site. Okay, so this is how I would choose typically. And then if you have, for example, multiple experimental structures that are of similar properties, for example, let's say they do have the same resolution, they're not missing any structure. What I would look for example, if one of the structure is for the protein alone and the other structure, for example, is for the protein in complex with the ligand, So because definitely in this case, I would compare both to see if there is any conformation change that is occurring in the binding site of your protein. And in this case, I would favor, for example, the one that is in complex, for example.
And in case there is no template, there is no structure. So
Okay.
I guess you have to choose the predictive modeling to have
Of course.
a model. And so how do you choose the template for that?
Okay, so the choice of the template. So if you're going to go for a comparative modeling, usually you will have a list of criteria. So for example, you could look for a template that shares a high sequence identity with your query, a high coverage, if you're going to go for a comparative modeling, for example. Or sometimes you can do what we call a multi-template modeling, Lilia. In this case, you're not only using one template, you're using multiple templates. And this is, for example, how, so this is something that you can use, for example, in Modeler. You can do the same for iTaser. If you go for ab initio, which in fact could be more challenging because you're limited typically with the size of your protein. But let's say if you look for AlphaFold, for example, AlphaFold is going to start by collecting a long list of possible templates that is going to align with each other, try to extract motifs, and sometimes it could even use structures that already exist in the PDB in order to improve your prediction. Of course, all this using deep learning approaches. In this case, again, there is no direct answers on the best choice of the template. It's case by case, but as I said, it's usually you have to favor the sequence identity and the coverage usually. And then based on that, you can choose, some of the templates or even go for multiple templates, predictions.
Okay, one more question, please. So I'm curious, and do you think that ligands or molecules that are identified as potential ligands for models are as good as ligands or molecules identified with real structure?
Okay, so your question, if I understood it correctly, it's typically comparing the results, let's say, of docking if you do it on a predicted versus if you do it on an experimental structure. Is this
exactly?
Okay. Well, it really depends on the quality of the structure, independently whether it's an experimental or whether it's in fact a computational structure. So we do have different tools that allow us, in fact, to assess the quality of the structure that we're working with, and based on that, in fact, it will allow us to be more confident or less confident in the model that we are using. So for this reason, in fact, if someone is going to just tell you that, okay, I use a certain approach to predict my model, in fact, and there was no validation steps of the structure's quality, definitely you cannot really trust the results. So that's why you have different approaches coming from ProCheck, Prosa. Let's say in AlphaFold, they have their own metrics to give you an idea on the quality of the structure that is being predicted. So definitely, if your binding site now is typically in blue or in dark blue, it means you can trust the predictions in this region of your model. If it is, let's say, in the yellowish part, in the red part, in this case, the confidence of your prediction is not that high, and in this case, it's not even worth taking the model to drug design or docking. It could be useful for other approaches for structure, but not really in the drug design part, because this is where you're expecting, in fact, a good quality structure, whether experimental or predicted, so that you can trust the results. Of course, keep in mind, like any other approach, you always have those what we call false positive, false negative. So when you're doing, for example, a screening of a large library of ligands, you're always you will always do certain mistakes, right? So you will be skipping, or you will be in fact rejecting certain promising drug. So in this case, you're dealing with those false negative. It means your approach or your high throughput approach didn't really identify this drug as being promising for your target. But on the other side, you could have as well the false positive. It means you could include a certain drug that is not really important, or it's not really interesting, or it's not going to show any promises later on in the process. But all these, in fact, structure-based drug design approaches, in fact, they play a very important role, and this is what I call, in fact, the enrichment process. So what you're trying to do in this case, you are trying to increase your percentage of good potential compounds in your set. So if you're going, let's say, for those 200 million, for example, you could have, for example, 0.01% of the compounds that are interesting for your target. So by filtering those, even though you're doing certain mistakes, you will end up with a data set at the end that could have, for example, 10 or 15 promising drugs %. So definitely you are enriching, your data set, knowing that you could be eliminating some very important compounds, and you could be including some false positive in your data set.
Okay, thank you. Thank you very much, Joseph.
All right.
Yes,
Thank
it's
you.
great. Any more questions?
If now, let's move to coffee break. So join us and do not hesitate to enhance discussion and network. So we have almost 20 minutes for that. Okay, you are welcome. Thank you, Joseph.
Thank you, everyone. Have a fruitful workshop, and enjoy every minute. Thank you very much.
Thank you. Bye-bye.
Yeah.
Hello, everyone. So it's my pleasure to introduce our next speaker, Dr. Ahmed, who is an assistant professor of bioinformatics with over 15 years of experience. He is currently based on the Department of Genetics at Farhat Hashad University Hospital and holds a co-affiliation with the University of Witwatersrand Strand in South Africa. His research spans pharmacogenomics, clinical bioinformatics, biomedical text mining, biomolecular modeling, and development of computational tools. Dr. Hussein leads the Agora team project and advocates for reproducible research and data sharing in bioinformatics and genomics. Beyond his research, he also serves as a reproducibility coach and mentor for research in Tunisia and across Africa. So we look forward to your presentation. Please begin.
Thank you so much for this nice presentation. I actually sent you the text, so I just thought So so I think a lot of things happen now, and very interesting things are happening now in genomics, in pharmacogenomics. I think we've been the trend, or we were part of two generations. So 15 years from now, exactly in this same place, we were looking for data. We've been we're very happy if we have like 20 sequences or 100 sequences to analyze. And now we crossed the other realm of high throughput data. So really, it's a very interesting era to work in. And this same philosophy, or this same mindset, I'd like to present, or I will present the results and the input from three main projects and the efforts that we have been doing now since 6 years, which was initiated by Scott Hazelhurst at the University of Witwatersrand in the frame of H3 Africa GSK Admin project. And then there's the project Agora, which I lead, and also the project WMA led by David Twasigame, also at the University of Witwatersrand. So I'd like to share with you a framework or a culture that we have developed to study the pharmacogenomic landscape in Africa, but knowing also that what we have been doing could be extrapolated or could be applied in other problems. So this mindset or this framework is based on three pillars. First thing is, the first pillar is the genomic analysis. Mainly we do secondary analysis, which means that we take data from other collaborators. So we ask them for data, for NGS data, whole genome sequencing, or exome data, and we specify our problem. We want to study pharmacogenomics landscape. So they gave us the data, and we analyze them using bioinformatics. And then the second pillar is using Batiscape or biomolecular modeling. It's a fancy word for structural bioinformatics. So we do that to interpret the variants and to analyze the functionality of these variants in order to predict what is the outcome on the pharmacogenomic landscape. And the third pillar is the application of the AI-based bioinformatics tools. So namely here we developed two workflows or two tools. First one is SWOT, and the second one is DRINK. But it has nothing to do with drinking, actually. Why we study the pharmacogenomic and landscape? So we did that specifically for what we call the ADME genes. So these ADME genes are involved to the absorption, distribution, metabolization, and excretion, which are key mechanisms, physiological mechanisms that will determine the fate of a drug within the organism. And here I'm just giving the picture of the protein CYP2D6. It's a main metabolizer for a lot of drugs, and so these types of proteins are highly polymorphic, and they could be this polymorphism could change from population to other. So which means that within, for example, the Tunisian population, we have a specific genetic variability. So it's very interesting to know this genetic variability, because it will determine if a drug will work or will not work for specific disease. And the number of the genes, it's about 300. You will find also some reports that will say that these genes are actually 340. So it depends on who you ask. And they are composed of basically 32 core genes. These are very important pharmacogenes, and also 263 extended genes. So in order to study these genes, we started, like I said, collecting the data, and we had a very nice data set. Actually, we have more than 450. We have 1,000 because we included also the 1,000 genome data set, and we collected the data from almost the 4 corners of Africa, except for Algeria. We discarded that because it was one sample, but we had from Southern Africa, Eastern Africa, and East and Western Africa. Most of the samples, they came from Southern Africa, which is good. And we also included some samples from Khoi and San populations, which are based in Namibia and Zimbabwe. Okay, also from South Africa. So the first thing we did is to subdivide these populations using not PCA, just a projection, but a clustering method. And we have identified 4 main groups: Southern Africans, the Far Eastern African, Western African, and the South Central African. And you can see here there are some outliers. These represent actually the Khoi and San population. So 4 groups. And the first work, or the first thing that we have noticed is that this genetic variability consists of mainly of rare variants. And this work was mainly done by George Darusha. So for the 4 groups, the Southern African, you find, for example, 133 unique rare variant. For the South Central Africans, 109. So most of these variants, they are rare and they are unique to each population. Okay. And then also we have analyzed the geographic distribution of these variants, and we found that actually the genetic diversity of these variants, it does not really tell about geographic proximity, which means that for each population, you have a specific set of variants that will determine the pharmacogenomic landscape. And this is expected from Africa, of course, because Africa is the most diverse population in the world. But then even for populations that are very close in the geographic proximity, it does not really tell that it does not correlate with the genetic differentiation. So what does that tells us? It's very actually very straightforward. Basically, if we increase the sequencing in Africa, we will discover more rare variants. And these rare variants actually, so for example, you can find some unique rare variants that will not have a fate or will not have an impact on the functionality of the ADME gene, but also you will find some other variants that will have the impact. And that really have an impact, an economical impact, and an impact on the way that to perceive the pharmacogenomic pharmacogenomic research and pharmacological research in Africa. So, for example, in the case A, if we have a subpopulation, so here, let's say, for example, we only sequenced 50 individuals, and we've been able to uncover 2 groups. So each one of these groups could be assigned to one drug. So this drug will work with this group, and this drug will work with this group. Okay. But then, if you increase now the sequencing, probably you will identify also individuals for which you will find no alternative. Okay. So this is really will impose a lot of challenges, because you are actually requested to provide drugs for each one of these populations. Okay, and apparently, because Africa is so diverse, then this problem is really critical. The second thing also to think about is that if we think about the diversity of the drugs. Okay, so for example, if you have one protein, and for this protein, you have this is also highly polymorphic protein. So you can assign, for example, a drug, but it will work for certain variants. But for the same protein, if you have another variant, that drug will not work. Okay, so this is really also that can tell the complexity of the pharmacogenomic landscape in Africa. So 2 things: we have a very diverse population, and we have also the diversity of the drug. Okay, and really this creates lots of challenges. So it's created by AI. So the challenges are because we want to interpret these variants. So interpreting the rare variants in batch is challenging, because we want to interpret everything in a high throughput manner. Okay, and if you want to do that experimentally, good luck with it, because you have to do functional analysis, you have to probably express proteins, and also we have a lot of challenges in low middle income countries to do that. Okay, you have to get some big infrastructure that could be very hard to implement in order to do this sort of analysis. Also, existing approach, they lack the cognitive model. When I say cognitive model, is the theoretical model, because as biologists, as scientists, we want to understand things at the fundamental level. So most fundamental level is the molecule. So it's very, very informative if you can see the molecule of DNA, for example, the 2 strands of DNA, because it will tell you about the replication, it will tell you also about the central dogma, it will tell you about everything. So having this cognitive model will help in interpreting any biological phenomenon. Okay, same goes also for variants in rare diseases. So we want to have this perspective, and the existing approach, even empirical approach, are very hard to implement and could not sometimes also give you this valuable perspective. And fourth, the current in silico approaches are not suitable for PGX studies, which means that these in silico approaches are tailored mainly for generic cases, diseases actually, or proteins or genes that if you modify something in these genes, then you will have let's say a deleterious effect. But for pharmacogenes, for example, it doesn't work like that, because if you touch something in or modify something in a pharmacogene, could, for example, not working for a drug and will shift to another drug, for example. Okay, so the evolutionary perspective is not the same for pharmacogenomic for pharmacogenes. So we could operate in 2 manners. The first manner is doing a global screening, which means that we screen all the genome and to identify the variants, and then we will have to take each one of these variants and say that this one is going to work with this drug, this one is not going to work, this one will interact with a certain drug, this one is going to modify the behavior of a protein, all the protein dynamics. Okay, and then the second approach is to do that by gene-wise screening. Okay, so gene-wise screening, which means that we will focus on specific genes and we'll do an in-depth analysis for each one of the genes. Okay, so Now, if we want to do the first approach, global screening, basically those who are well versed in genomic analysis and GS analysis, they will use certainly what we call the variant effect prediction. So variant effect predictions are actually very biased methods. Okay, so they are lacking poor. They have a poor benchmarking, which means that they are not well tested. So it's very risky to use them. They lack sensitivity. They also they use a very generic data set for training, because you apply also machine learning and AI methods in order to do these this type of analysis. Well, not you, but persons who implemented or invented these techniques. Also, they sometimes lack what I call also the molecular perspective. So they don't give you an overview about the molecular mechanism. And also, if you want to use multi-scale modeling, such as molecular dynamics, well, you can do that also in screening. You can just screen all the genome and do the molecular dynamics. Okay, and one of these tools is SIFT or PolyFin2, for example. So, how did we approach that? In order to interpret the variants of pharmacogenes, we invented the SWOT workflow. So the SWOT workflow consists on actually two sub-workflows. I'm showing here just one. This one actually takes the data from the database of homology models actually and crystal structures. And using this crystal structure, we will apply thermodynamic analysis, and also we will calculate some scores, and these scores will be combined, integrated together, in order to say that this variant will work and this variant is not working with this protein. Okay, and as you can see, maybe you are familiar with the protocol or the tool Foldix. So we use a tool that will calculate the thermodynamic properties, open the changing of the protein. This is the list of the protein that are integrated within SWOT. And as you can see, for this list, it's very, I would say it's limited. So limited for certain pharmacogenes. Okay. So if you are if you want to do annotations for, let's say, proteins for specific pathways, you have to do things manually. So you have to do the manual modeling. You have to extract the structure, create the structures, and then integrate that to our work with SWOT. But we have also very nice coverage of the main proteins that are involved in pharmacogenomics. We have the SIP families. We have also the UGTA, GSTM1, GSTP1. So yeah, we have a diversity of structures. And then within SWOT also, we have integrated sequence-based features. Sorry, so we have integrated a machine learning model, and this machine learning model was trained on specific features. So these features are sequence-based features and also the structure-based features. And as you can see here, for the structure-based features, they are very important to train the model and to show such accuracy. So we've been able actually to have an accuracy score better than most of the well-used tools in annotating the variants, such as SIFT, PolyFin2, ProVin, and so on. Okay. And then if you look to the performance of SWOT, both it's better than most of the other tools, except maybe for PolyFin2. But in some cases, actually, in some of the pharmacogenes, SWOT performs definitely better than any other tools, especially for cytochrome P450. And we tested that for DPYD. It's a very famous gene, ASMID gene, actually. But we also did some benchmarking for TP53, a generic protein. And if you want to work with SWOT, actually, you have to just download it from the GitHub repository. We'll actually have to do some modifications now, but you'll be able to just download it and work with it if you know how to work with Nextflow, for example. Now, the second tool that we have built is called Drink. So this tool actually, it will model the modifications in cytochrome P450 proteins. And the idea here is to integrate both the diversity in the sequences, which means the mutations also, or the variants, and also the chemical structure of the ligands. So what we did actually is collect the data set, the training data set, that you have the ligand, the chemical structure, and you have the sequence of the protein. And then we have built a classifier that can tell if the binding of a ligand with a cytochrome P450 protein will be strong, or medium, or weak. And this work was done by Blessing Gutandra Sitabuli, my PhD student at the University of Witwatersrand. So here you see a matrix of features. We calculate a matrix of features. In blue, you have the scores or the features for the sequences. And in green, you have the features that were calculated for the ligand. And we've been able to calculate 27,000 times 30,000 features. And then we did feature selection protocol, feature engineering, and we integrated that in the machine learning. So all this workflow was implemented with Python, and we used MetaFlow to do the implementation. Sorry for the next flow, guys. And the performance actually showed a very important and accurate score, especially for the XGBoost algorithm, naming, for example, the accuracy here was performed better than any other classifier. But then also we have tested some instances or some combination of certain features. We did apply also the compression techniques, such as PCA or autoencoders, and we compared them together, and we found that PCA actually is better than the autoencoders. Again, this work was done by Blessing. And if you see also the performance, the ROC curve, we found that the drink algorithm or tool did a good performance for the three classes that we tried to predict. Now, that shows real case, shows how to integrate the data in order to predict the fate of a ligand of the binding. Now, the other tool that we have predicted actually, or we have developed, actually does the text mining, or it predicts actually if you give it a phrase like this one, for example. So it does the tagging, which means that it will identify the drug and the gene, and the predictive model will tell you that this is actually a phrase that describes a pharmacogenomic relationship. Okay, so as input, we'll have an abstract or the generic test, and we'll subdivide this abstract into phrases, and it will extract the phrases that will describe the pharmacogenomic relationship. And actually, we trained the model based on abstracts from PharmaGKB database, but also this is for the positive relationship, and for the negative relationships, we took the random text from PubMed, and we have actually mirrored the text from PharmaGKB in order to reproduce or to decrease the bias. So the featurization of these steps was done using the TF-IDF vectorization process, and then we did all that we built the model using support vector classification models, and we did the validation on 20% of the data set. And as you can see here, the ROC curve, it's almost perfect because you have AUC 0.99. Okay, but life is not fair actually. Why? Because if you look to the ROC curve, you may say that, yeah, this is too good to be true. And it's actually the case because the model actually will tell you or will give you some phrases that combines or that returns a relationship of certain genes from other animal kingdoms actually. So we had, after applying the model, we had to do a manual regression, which identified 296 high confidence drug gene relationship, and 127 actually relationship for malaria, and 170 relationship for actually TB, not malaria. Sorry for the mistake. And using that approach, we identified 24 pharmacogenes that are involved in TB and malaria pharmacogenomics. And from among these genes, we have identified 9 less known genes. So these genes actually, they weren't really known in the pharmacogenomic realm to start with. And we use this data actually to do the prioritization. Sadika actually did the genetic characterization of some of these genes. And as you can see here, for Africa, with the dark blue bars, the genetic diversity is very high compared to other subpopulations like South Asians and the European populations. So yeah, we use that model actually to guide our analysis, our genetic analysis. Now, the other thing that we did is done by Nklamulu Khuza. It actually does the study of what we call the haplotypes or the star alleles within these pharmacogenes, because the genetic diversity doesn't come always with single variants. So you have a block of variants that segregate together, and we applied a method called normal mode analysis in order to predict the fate of the variants. So thanks to this model, actually, we actually combined the entropic term and also the thermodynamic calculation of the enthalpic term, and we've been able to predict also some unknown haplotypes, or the fate of some unknown haplotypes, using this model. So this model is actually, or this method actually, is very cheap. It doesn't really require a lot of computational power. We did also the analysis of some of the pharmacogenes. So now we go into the molecular dynamics method. So previously we did the screening. All the methods I explained, they do the screening for entire genome maybe, but this one actually won't be able to do that only for a handful of variants, and that's why we applied the molecular dynamics method for 5 or actually 6 of these variants. And these variants are very rare and within the African population. And the question here is that because we don't have any functional data about these variants, we want to know if these variants could impact the cytochrome P CYP3A5 protein or not. Okay, so we did molecular dynamics, and actually we found that so molecular dynamics is simply putting the protein within a box of water, and you try to simulate the physiological conditions. And we found that indeed these variants actually could change the conformational behavior of the proteins. As you can see here, for example, this energy landscape. So you have here wild type or the reference structure, and compared to this wild type, you have all the other variants. They are above in terms of energy. They are above the reference structure. And when we tried to check that visually, we found that this actually corresponds to a slight opening in the channel in which the drug will go through. Okay, so really it explains that these rare variants, they do have an impactful effect. We did also the calculation of the volume of the pocket, and indeed we've been able to explain that these variants are actually responsible for increasing or decreasing this pocket. And sometimes it will work for some drugs, like here, but also sometimes it will squeeze the pocket. And this is actually a docking combined to molecular dynamics for 2 drugs. One of them is actually an anti-HIV drug, and the other one is the anti-malaria drug. Okay, so we've been able to prove that the behavior of these variants depends on the drug. And if you wonder, could you maybe apply these methods for other analysis, not only for pharmacogenomics? Well, you can do that. And actually, this is what we did before with Dr. Yusuf Hamdi. So we helped her actually to interpret a specific variation within the RAT50 protein. And actually here we didn't use molecular dynamics, but we used a cheap method for doing the dynamics of the protein. and we've been able to prove that the interface by which this interface here, by which the protein is interacting with its protein partner, is actually changing between the wild type and the mutation. So we've been able to do that, and definitely you can do that also. So what's next? We are now testing the AlphaFold algorithm to integrate it to these tools like the SWOT, for example, because in SWOT, like I showed earlier, we used only a handful of protein structure in order to do the annotation. But now with the upcoming of AlphaFold, we can do the screening for a very rich set of proteins. Okay, and the work done by Ayman Blade, it shows that here you have, for example, a comparison between AlphaFold and crystal structure. Here you have comparison between AlphaFold and the experimental structure and the homology model. The homology model and experimental structure, and AlphaFold and experimental structure. And you can see here that the correlation is almost 0.6, which means it's very similar between the three methods, which is, I won't say it's a good thing, but it's not also a bad thing. So it shows that the behavior of the thermodynamic calculation maybe does not depend on the structure that we use, which means that AlphaFold could work in this case. And the final case here, this is a pharmacogene that was calculated, or the data were calculated by UNA. I don't know if you see the points, but you have almost a perfect match between the AlphaFold algorithm and also the cryo-electron microscopy structure. Okay, which means that probably AlphaFold is very useful in this case. So I have to be critical for AI methods. We have to do that because we found a lot of hype and everybody's talking about artificial intelligence. And the thing is that to date, AI applications in pharmacogenomics are primarily limited to isolated projects, like the one that we did, with minimal access to real-world data. And the second thing is that functional variant annotation has reached saturation phase. So almost we do the training on the same data, and we have almost all the classifiers, all the variant effect predictors, they're now behaving the same way, which means they're not really giving an additional value for genetics or pharmacogenomics. And in order to challenge that or to improve that, we will have to get access to a rich set of data, which means the data has to be collected systematically. So, for example, if a patient come to the doctor asking for treatment, you need to get these data and integrate them and use them to train the models. Maybe you can do a lot of things, and you may apply also some very advanced statistical models. And we have actually exposed all these problems in this review here. You can check it. I'll be happy to send you a copy. But so far, what we are doing actually is piling up the data right here at the discovery domain, which means they could be useful, but they're not that useful. And we wanted to cross to this domain here. So this domain, in order to get to this domain, we need to enrich the data. The other thing also, we have to question the input from low middle income countries in drug repurposing. Because why are you doing this workshop? To train people on drug repurposing, but we want these people actually to work in their country. So we need also to think about the economy. We need to think about the facilities, how to render this environment competitive to any other companies like GSK and Novartis. So why I'm doing the advertising for GSK and Novartis? Because they funded our research. And I'd like to thank all my team, my Couscous team and my Bright team, South Africa. And if you are interested in seeing all this list of papers, then you can just have this QR code, and I'm happy to receive your questions.
Thank you very much, Hussein, for this insightful presentation. And now let's open the stage for questions and discussions. So please do not hesitate.
I don't understand the last slide from precision medicine.
Well, actually, what like I said, what we are doing so far is like having a small set of data in isolated cases and very specific cases actually. So maybe you are interested in a specific system that you want to improve the annotation process for this specific system. I know when I say system, could be a protein, could be a protein family, for example. But if we want to democratize the usage of AI in drug repurposing, in pharmacogenomics, everything, it has to be integrated within a larger ecosystem, like in clinical record system, which means that the workflow has to cross from the hospital. You have to do maybe a deep phenotyping. You have to collect everything about the patient, including the size of his shoes, maybe, and then you can integrate all these data to using advanced statistical modeling. Given also that you will have to get access to some high performance computing facilities, and you do the discovery phase, and then you can later implement in order to cross to the actionability domain. Means that you have to validate what you find to do the to identify the specific interventions within the clinical ecosystem. Yeah.
Thank you again for your nice talk. I have a question regarding your first work that you described,
Yeah.
Regarding the landscape, pharmacogenetic landscape in Africa.
Okay.
And I'm wondering why rare variants.
I think the possible cause here is that because normally we tend to look at the population in Africa as a homogeneous population, but the reality is not the same. So, for example, we have few data now for Central Africa. In Central Africa, maybe it's a rich forest environment. It's exposed to a rich forest environment climate. But then if you go to Eastern Africa, it's totally different. Okay, so the way that we eat, the way we behave, the climatic also changes. In Southern Africa, for example, we have also more ancestor population, like the Khorasan, for example. So they are differently submitted to different evolutionary pressures, and these evolutionary pressures they tend to modify the pharmacogenomic landscape, and this is why actually we are seeing these rare variants. So, which means also that if you found a drug that is working for a handful of people in the Khoisan population, it won't necessarily work for Zimbabwean population, which is very close to Namibia actually. So geographic proximity does not mean genetic proximity. Yeah, especially for pharmacogenomics.
So may I add? Thank you, Hussein, for the script. So my question, it's nice to have the genetic landscape of pharmacogenes in Africa. And do you think that a custom array to facilitate the genotyping of these variants could be developed and used in a medical setting? Is it realistic? Otherwise, why do we do such studies?
So this is a good question because the cost of sequencing now is still high. For either it's a genomic whole genome sequencing or whole exome sequencing. If you
genotyping array.
Yeah, for genotyping array now, if you do that, you have to use maybe an imputation panel. So if this imputation panel is proved to be reliable for, let's say, the Tunisian population or the South African population, then what you can do is you will just genotype your individual, and then you do the imputation, and whatever variant that will be discovered later, you can validate that using standard sequence. It won't be as fast as the genomic sequencing, but then the cost, actually the cost of the array could go from $70. If you do the exome now, it's $400 to $500. So for Tunisian patients, it's very interesting. But even at the fundamental aspect, also fundamental level, it's very, very interesting to know the structure of your population first using a microarray before going to sequencing your population.
Okay.
Thanks, Hussein, for this nice presentation. Just looking at this Venn diagram.
Yeah.
So, did you try to look at the effect of these specific variants for South African population, for West?
Yeah. So not specifically. Yeah. So actually, we did that. We did that but only for a handful of genes, not for all genes. And indeed, we found that there is a big possibility that these variants will impact the fate of the drug. And this was done not only on cytochrome P50, it was done also for transporters, for modifiers. And each time we take randomly one of these variants, it appears like visually, and when I say visually, using molecular visualization, for example, not even calculation, we find that a variant that is very close to an interaction site or a catalytic site, that could really impact the fate of the drug. Yeah.
And also it may be explained by the sampling. Number of samples?
Yes, for the sampling, for the sampling, actually, we had a problem with one of these groups, actually, the pharmacists.
Yes, I see.
Yeah. So that one, but for the other ones, actually, we have.
And the variant array, according to public databases, or are rare in Africa.
They are actually rare. Yeah, they are rare compared to what we identify as populations here. But even in comparison to 1,000 genome or normal data or anything, still rare. But also they don't have sometimes they don't have the same frequency in these populations. So we found maybe a variant that is more prevalent in European population or South Asian population, but it's not that prevalent in African populations.
Okay.
And actually, we thanks to these data, there's one other work that haven't shown. We've been able to point to the problem of using hydroxychloroquine in Africa at the time where Donald Trump asked everybody to take hydroxychloroquine. So actually we've been able to point to possible toxicity, especially for population in Western Africa, using these data. So genomic data, but also array data, genotyping.
One more question?
Yeah.
Okay. Thank you, Hussein, for sharing this great work. My question is about the SWOT project.
Yeah.
Is it a prediction tool based on structure, protein structure of the effect of the variant of the structure of the protein?
Yes.
So the input is the VCF data.
Yeah.
And the output, like there is thousands and millions of variants from NGS. So the output is only the pathogenic predicted proteins, right?
Okay. So the way it works, you give it a VCF file.
Yeah.
It doesn't matter if you give missense variant or not.
Yeah.
So wherever variant that maps to the protein structure, it will figure out in the reporting later. Because specifically for SWOT, we have some protein that will cover we have some structure that will cover all the protein, but we have also some partial structures.
Yeah, this is
exactly.
And
this is actually why we're moving toward maybe applying AlphaFold algorithm in order to integrate it into SWOT, but not only the modeling part, not only like giving the full protein to the workflow to work with that for the annotation, but we will change maybe FoldX to another tool. It's called DBBGAN. Was shown that DBBGAN in the benchmarking, it was better than FoldX, and it's subjected to less bias. So we will integrate that to the workflow. but also maybe we will get rid actually maybe for the VCF file.
Okay.
Because what we noticed is that there is a confusion between the 37 build and the 38 build. Because I haven't shown also the other workflow, the annex workflow that will do prepare the database for you. And I think now we don't have any tool that will automate that in an efficiently in a computational efficient manner. So instead of using the VCF tool, a user could just give the position of the protein compared relative to 38 build or the 37 bit, and we give you the annotation results. Give you also the belonging, if it's going to be deleterious by it or not. Give you also the report of energy and many other physical chemical properties.
So just to clarify, it can't predict the effect of the variant on genes that are not, they don't have a PDB structure.
Exactly. Yeah. So what we only integrated some dozens of these proteins here, but also we allowed the user to integrate wherever protein wants to manually.
Okay.
But it's not. I have to admit it. It's not a very straightforward process. So now we're moving actually toward improving these the ergonomy of the workflow, so that any user can deploy it in a very quick way.
Okay. What distinguish between, because there are other predictions that are based on the energy of the structure of the protein. So what's the advantage of SWOT?
So at the time of the development, the advantage was that there weren't a lot of tools that do the genomic the variant effect prediction from genomic data. Now there is also well, the AlphaFold, of course, but there is also the alpha missense score that was built based on AlphaFold algorithm. So we think that there is a tendency to now annotate genetic variants based on protein features, but specifically for SWOT, it does that not only on protein features, based on protein features, but specifically on thermodynamic features, which wasn't observed a lot in many of the tools. Even of the tools that can do the genetic annotation based on protein data, they don't really integrate the thermodynamic features. So delta G, for example.
Yeah. Like I mutant they check for.
I mutant will give you actually you have to specify the coordinate of the mutation for the protein.
Yes, that's true. Yeah.
Yeah.
Thank you.
So,
We have one more question. Yes.
Yeah.
Thank you for your presentation. So my question is, you've tested a lot of machine learning algorithms.
Yeah.
So I'm wondering if there is any algorithm that has an exceptional results, but and if you test the deep learning algorithm.
okay, so I think Joseph talked about that before. The keyword for this workshop is that the only one golden rule is that there are no golden rules. So nothing will work with everything. And you find we found maybe me and Osama later, we're trying to avoid to answer directly your questions. What is the best tool? Because that's actually what happened. So for example, in the case of SWOT, but even in the Brink tool, we didn't find a good performance using the deep learning algorithm. But that could be also because we haven't introduced a lot of complexity of setting, okay? And this was intentional actually, because this tool is meant to work on with sample resources, because not all the users, they will have the access to GPUs, for example. So sometimes we prioritize the low complexity with some sacrifice on the accuracy scores or anything, but this tool could be useful also for a lot of people. Okay, we did that with Brink. So Brink was already like deep learning algorithm wasn't the best performance, but then for for SWOT, that was our philosophy when we started to develop that tool, which means that it has to be deployed in resource limited environment. Yeah.
Thank you very much for
But the problem when you have the correlated feature, what do you use? Not the machine. Machine doesn't work with the correlated feature.
So we have actually we washed out any correlated features. For example, for drink, we did the filtration for the sequence-based features, but also for the ligand-based features. So for more specifically for the sequence-based features, because we relied on the AA index database. First thing we did is that we keep only features that are not correlated. We end up with 60 features actually, from a total of 500, which was good actually.
But when you pick the correlated, the
So-
result is not.
If you filter the correlated, it means that you remove the redundancy, because why keeping two things that will say the same thing?
Yeah.
Okay, so because if you include the correlated features, it means that you will increase also the calculation time. So yeah.
When you use the machine learning, what is the input and the output in the lesson?
Well, good question also. So the input it could be a CSV file. For specific for the drink algorithm, it's the CSV file. You can give the position of the mutation and the name of the gene, and then you give also the smiles of the ligand.
Specify some
Yeah.
parameters.
Smiles, which when I say smiles, Usama will show you later. Smiles is just a way to encode the structure of a ligand or a drug. Okay, so you give it that, and in a CSV file could be also an Excel file, and then it will process that to give you the outcome. For SWOT, it's a VCF file, which is basically also the
There is a step of the preparation for the DSP file, or that's we put the DSP file. Yeah.
So for the VCF file, we need only to specify the coordinates of your variant. Yeah.
The limitation.
Yeah, it is working on 37 build, still working on 37 build, but you can do that very easily with a tool like leftover or something. But for the other, for Dream, it's just you can use spreadsheet or Excel. So yeah.
And the output.
So please, you can continue after during the break. Yes, we have 1 hour.
Thank you.
Thank you.
Now we will begin. So now we have the honor to present Dr. Walid Gehat from Egypt. So it's my big pleasure to introduce you, Dr. Walid. So as Dr. Lia said, Dr. Walid is the partner of Dr. Lia during this project. And Dr. Walid studied pharmacy and obtained his master's degree in pharmaceutical chemistry at the Faculty of Pharmacy at University in Egypt. And then he was awarded a scholarship from the German Academic Exchange Service for doctorate study in Germany. And in 2016, he got his PhD in medicinal chemistry under the supervision of Prof. Rolf in Saarland University. He then returned to Egypt and was appointed as a lecturer of organic chemistry at the Faculty of Pharmacy. And in 2017, he joined the group of Prof. Anna Hirsch as a postdoctoral fellow at Helmholtz Institute in Germany, where he is currently leading his junior research group. He is also focusing on the application of modern drug design approach, tackling under biophysical and computational techniques for drug discovery and repurposing. So, without further ado, so we look forward to your presentation. So, please begin.
Many thanks, Ms. Lee, for the kind introduction. Also, thanks for my dear Lilia for the kind invitation to be here in Tunis and share my research work with you. We will talk about the drug design using computer-aided approaches to tackle new diseases and also rare diseases. So my first question, do we need new drugs? Yes. No. Excellent. Exactly. So why? So of course, there is always emerging new diseases. We all witnessed the COVID pandemic and no antiviral drug could be suitable for this, right? So another reason, the drug resistance. So you are also working in infection diseases. Bacteria, viruses also always mutate and get resistant to the antibiotic. The same actually in human cells when they are malignant. So the chemotherapy also sometimes getting resistance. Okay, so we need new drugs. Lack of effective treatment. So until now, some diseases still cannot be completely cured. The Alzheimer's disease, rare diseases as well. And also, for some therapeutic indications, the available drugs have narrow therapeutic index. This means the margin or the difference in concentration between the toxic dose and the efficacious dose are narrow. And we should be very careful when monitoring the drug concentration to the patient. Otherwise, it got into toxicity. Major examples are the antidepressant and antipsychotics. That's why actually we need new drugs for these diseases, more safer drugs. And a recent study actually indicated that, or estimating that the number of deaths due to major diseases expected to be increased from 25 million up to almost 50 million by the 2060 due to lack of efficacious drug treatment for certain diseases. Whatever the status of the country, if it's high income country or middle income country or low income country. Okay, and here are the list of the most prominent diseases which are expecting to be still escalating with reflecting the need of new drugs. Furthermore, the high cost drugs, for example, interleukins. We need to find new, cheaper drugs for the low income countries, right? And finally, sustainability. So these pharma companies, big pharma companies, hire 100,000 of people. And if they stop producing drugs, okay, or developing new drugs, and after a time, the patent will stop or will be expired. So then how these companies would be sustainable? Okay. So as presented in the morning, the pathway for developing new drug from the scratch actually is quite long. So it takes from 10 to 15 years. And we can focus on the first stages here, stage 1 to 3. It can be divided into the target discovery and validation to know the causative, the molecular level of the disease in human responsible for the disease, and then finding the inhibitor or the binder that could modulate this target and treat the disease. And this stage of drug discovery and development can be subdivided into hit identification, hit to lead, and lead optimization. And if the compound is successful on in vitro level, then it can be transferred into the preclinical studies where you test it in animals. We call it in vivo, and check if the compounds also efficacious, not cytotoxic. And if pass all this, it will go to the clinical trials when you try to test it in humans, and so on and so forth. Very importantly, computer-aided drug design or in silico drug design actually can help us all over these stages. For example, in the target discovery and validation, we can apply reverse docking, protein structure prediction, the homology modeling, and target druggability. for hit identification, virtual screening approaches, de novo design, pharmacophore, similarity search. For hit to lead and lead optimization, you can do structure-based optimization and 3D QSR, quantitative structure-fit relationship. And even for the preclinical studies, where the pharmacokinetics, pharmacodynamics come to be a major rule, you can also make prediction models for the admit production properties and also models for pharmacokinetic dynamics and toxicity. Okay, so in the next slides, I will show you some highlights from the work where we applying the in silico approaches for understanding the drug interaction and discovery of new drugs. So the first case here is structure-based design of dual bacterial RNA polymerase and HIV-1 reverse transcriptase inhibitor. So do you know when the World AIDS Day?
Today. Yesterday.
Excellent, exactly. So this is actually relevant to what's going around. So HIV is a human immunodeficiency virus, is a causative agent for the disease, acquired immunodeficiency syndrome, AIDS. It affects 40 million people at the current estimation, according to the WHO, and with 1.3 million people in new infections in 2023. So it's relevant and dangerous issue for us. HIV affects actually the immune system, our immune system. This means, and this is our defense against bacteria, against viruses, and so on. This means if the immune system is weak, you are easy prey for these microorganisms. And that's why HIV patients are highly vulnerable for bacterial co-infections, such as MTB and Staph aureus. For healthy people, not sad people, we live with Staph aureus. It's on our skin and so on. But if you get the breast or patients like HIV patients, they are suffering from sepsis and other consequences of Staph aureus infections. Okay, so for HIV people, they also get several antiviral drugs. And if they are compromised with infections, this means you need to give them also the antivirals. And this could be problematic in terms of resistance development. So bacteria would make resistance, the virus would make resistance. So you need more antivirals, anti-antibiotics, drug-drug interaction, which somehow also mentioned in the previous talk, and complexity of regimen. So patients had to take 3 pills, 4 pills each 8 hours. You just need 1 pill and that's it. What we call the patient compliance. It should be friendly to the patient to keep taking the course of treatment. Okay, so based on this, we thought of developing our new antibiotics targeting the bacterial RNA polymerase into dual-acting antibacterial and antiviral drug. The rationale behind this was the similarity between the binding pockets in the bacterial target, the bacterial RNA polymerase, and the viral reverse transcriptase enzyme. So here, there is a new binding site called the switch region. It's like a hydrophobic pocket responsible for the flexibility of the beta clamps of the RNA polymerase. It's hydrophobic, and if it's blocked, then the free movement here for opening and closing would be hindered, and the RNA polymerase cannot accommodate the DNA. The same mechanism and the same hydrophobic structure is true here for the viral reverse transcriptase. So the viral reverse transcriptase has a thumb and finger domain, and this binding site inhibited by nevirapine also responsible for the plasticity of the binding pockets. So we thought, since they are mostly hydrophobic, and of equal size, and the mechanism is the same, we saw that our inhibitors for bacterial RNA polymerase could also be suitable for inhibiting the viral RNA reverse transcriptase. So we started with molecular docking. So here you can see the compound in magenta color superimposed with nevirapine in the binding site of the virus. And actually it adopts advantageous binding mode where no interaction with the amino acid residues which are frequently to mutate. Nevertheless, there is a space above the phenyl ring here, which you can also address by adding one more substitution, which could make more interaction with the protein and increase the affinity to the viral target. With this, we started to introduce different para substitution at the phenyl ring. And as you can see here, compared to the naked phenyl with 1.2 micromolar inhibition, the IC50 for the viral reverse transcriptase, by introduction of fluorine, the activity was increased. And then we were testing which substitution could be more suitable, and by increasing here any hydrophilicity, activity was reduced, increasing the size a bit, activity was decreased, and then further improved until the methyl sulfonamide group reaching 0.1 micromolar, similar to the reference compound nevirapine, and even much better than the other antiviral compound, the zidoflutin. On the other hand, the activity against the bacterial RNA polymerase was not reduced. To check this on the molecular level, you can see here that the introduced group here actually has been involved in more hydrophobic interaction with the tryptophan. And also it makes sense because if you introduce larger phenyl group here, as in the case of this compound, compound 19, you see directly a reduction in the activity, 9-fold reduction in the activity because there is limited space here. And compound 18 was the most active regarding inhibition of the viral reverse transcriptase. And furthermore, we tested the compound on different HIV-resistant strains against nevirapine, so 4 resistant strains here. And as you can see, the IC50 value, and these viruses are having multiple resistance, and the fault in the IC50 value is described here. So in contrast to the nevirapine, showing more than 200 increase in the IC50 value, the compounds keeping their activity against 2 resistant strains. Also, we investigated the mechanism of inhibition of the reverse transcriptase, which is mainly allosteric, and using the FRET assay. So the FRET assay, you measure the fluorescence by adding 2 fluorophores at the 2 ends of the clamp here, the thumb and finger domain. And if the distance increases, so opening of the clamp, this leads to increase in the FRET signal or the fluorescence. And if the signal decreases, this means the clamp is closing. And in contrast to nevirapine, which leads to increase in the intensity of fluorescence, the new compound acts via new mechanism via closing of the clamp or jamming it into a closed conformation. So this work, due to its importance, actually was chosen for the cover of Journal Medicinal Chemistry a few years ago, and was also had a lot of press release and praise in the media. If we don't have the crystal structure of the protein, the second option would to generate a homology model. And as also mentioned in previous talk, so there is several methods. The most reliable one to use a template, using a known crystal structure of highly homology protein or highly sequence identical protein. This would be giving more reliable homology model, or using AI tools which are faster and so on. So here, the basics or the concept of building a homology model, that So it's generation of protein 3D structure from its amino acid sequence. Why it's possible? Because protein with similar sequences are likely to have similar structures. This is by nature. It's not our theory. It was observed that the protein cannot adopt uncountable number of structures. It has limited number of structures. And the groups of protein which has similar sequences tend to have these similar structures. Great. So, yes, this was applied on a membrane protein called the pantosine energy coupling factor transporter protein of Streptococcus pneumonia. So the issue with membrane proteins that they are difficult to crystallize because they tend to be less solid. And this is a challenge for crystallization. That's why there were little known or experimental data regarding this protein. And we were interested for the protein from Streptococcus pneumonia. We applied the homology model, taking the template from Lactobacillus delbrueckii ECF protein, and build the homology model, as you can see. And you can validate if this model actually reliable or not by checking the RMSD results, the root mean square distance results. If they are low, exactly. So these numbers, for example, the alpha unit and the two alpha and sorry A and A prime units here showing the most lower RMSD values, whereas the substrate domain here showing the highest RMSD value. In total, this is of 3.2 angstrom, which is still acceptable. This homology model can be used for identification of potential binding sites. So you need to identify binding site if you want to do virtual screening, right? Okay, so it can be used for identification of binding sites and design of new drugs or optimization of existing ones, if you know the binding site. The third application is molecular similarity analysis. So this is also based on the concept or the theory of similarity probability principle, which says similar structural features give rise to similar properties, biological activities. If the molecules are the same or similar, it's expected also that they can interact with the protein with a similar method or similar way. Okay. Perfect. How to run similarity analysis? First of all, you need an active molecule. Your drug, you want to check if the database contains similar compounds that could have a similar activity to my drug. So you need reference drug. And this drug will be characterized in terms of fingerprint of the drug. This fingerprint now expanded to be hundreds of fingerprint methods. It could be due to the chemical connectivity of the molecule. So here we have nitrogen should be connected to sp2 carbon and the other side sp3 carbon, and the second atom connected to nitrogen from the right hand side and another carbon from the hand side, and so on and so forth. As this fingerprint would be presented in a binary strings or bit strings, 0, 1, 1, 0, and so on and so forth. Excellent. So the bitmap fingerprint, it's a connectivity method of describing the molecule, fingerprint. And the GBI DBH3, it's a pharmacophore fingerprint. So what we have here, hydrogen bond donor, aromatic interaction, hydrogen bond donor acceptor, and so on and so forth. You can select whatever fingerprint you wish from the software. Excellent. And then you will do the same on your database of chemical structures. Could be the drug database, could be commercially available database, the zinc, and so on. This, we have a nice list also presented today in the morning, and generate the strings for every molecule here using the same fingerprint. And then you compare which compound showing similar features to the string of your query compound, right? So what you expect here, Exactly. Thank you. Yes, but it's not by expectation. You need a mathematical method. Of course, I think you are expert in this. So we use the Moto coefficient. You heard about this. Okay. So to compare the binary bits of the compound and the database, you need a similarity metric. And there are many also. The most common use is Tanimoto coefficient, and also there is dice coefficient and so on. So Tanimoto here uses this equation. So C is the common features between the compound, the reference compound, and the compared compound, divided by the overall features of the reference compound, the other compound, minus the common features. And with this, the values of this coefficient would range from 0, meaning no similarity, until 1, meaning maximum similarity. Okay? Exactly. So, yes, correct answer. Excellent. So, we applied this on a actually very good project using sustainable materials. Starting from lignin, so the product and the waste of wood, and it was hydrolyzed to end up with nice intermediate here, which can easily derivatized into valuable compounds. And of course, since it's coming from wood, it's highly sustainable. And as you can see here, this building block or intermediate can be derivatized into alcohol, aldehyde, and then this can be used for synthesizing natural compounds such as dopamine, tetrahydrobiopyrrolene, or also can be changed into different heterocyclic compounds of potential biological activities. If you have a library of these compounds, you are not sure which activity you should screen. It's not target-based or project-based. Okay, so at the lab, we have facility for cytotoxic screening every day, antibacterial activity screening, routine work. So we did this for the whole 40 compounds, but we can also explore beyond this activities. So how to make use of in silico screening and similarity search. What we did, to use these compounds representative of each class of the compounds, for example, the amino alkyl guaiacols, the BERT, and use them as reference and compare them with the drug database for similarity and see if there are some hits with high similarity. Okay, so we use the drug central database. It's free, you can use it. The fingerprint, both actually, one chemical connectivity and one pharmacophore, and the metric Tanimoto coefficient, setting the range between 55 and 80%. Okay, and as you can see for the first class, we have some hits here. For example, dopamine, isobrinaline, and this is the similarity, and this is the activity and the indication. So we have antihypertensive drugs, COX inhibitors, this compound infenamic acid, and other indications. We did the same for each class of heterocycles synthesized, as I mentioned. Again, here we have acetylcholinase inhibitors, kinase inhibitors, anti-cancer compounds. That's also true again, COX inhibitors. So similarity for this compound, the clopirac compared to this class, and so on and so forth. And also here maybe so similarity 60% with norfloxacin. So these compounds could be antibiotics, and also here this compound could have antiviral activity. You should validate these results. So all these theories, nice, great, but could be also wrong. So we selected two activities, so biological activities, the cyclooxygenase inhibition and the antiviral activity for the compounds, and selected some compounds and tested them. For the cyclooxygenase inhibitory activity, here we picked 10 compounds depicted here in the graph, and first screened them at 50 micromolar, comparing to the I think this is Silico. Sorry for the bad resolution. And yes, the compound here showing the highest inhibitory activity and determined IC50 for it in range of 15 micromolar. So indeed, these compounds could be potentially anti-inflammatory agents. The same actually for the antiviral activity. We selected 3 compounds here, first testing at 10 and 5 micromolar. And you can see here compound 5FF showing nice dose-dependent inhibition of the replication of KSHP virus. And this was also used for determination of IC50 values, showing IC50 around 17 micromolar, which is even stronger than the foscarnet non-antiviral drug against KSHP. Further application is to understand the mechanism of interaction. So it's not common that you got the crystal structure of your compound with the protein, right? You can also do docking, but of course, should please take care of what we mentioned in the morning. There are some limitation. You should be critical and inspect if these results are true or just predictions. Okay. So here, this actually great work. I like it too much. So this was an investigation of the indigenous human metabolites in our body. 3 isomers called itaconic acid, citraconic acid, mesaconic acid. They are signaling molecules in our body. As you can notice here, they all having the same molecular weight. All are alkene with just rhizomeric isomers of the double bond, or cis and trans isomers to each other. Nevertheless, each molecule has unique pharmacological activity. First of all, the itaconic acid, the first molecule on the left here, showing the strongest succinate dehydrogenase inhibitory activity, which leads to anti-inflammatory effects. So we wanted to understand this on molecular level. So we looked the molecules and compared them to the oxaloacetic acid. So this is known inhibitor of SDH, and this is a crystal structure. And as you can see here, the itaconate in yellow adopting more or less similar binding interaction like the oxaloacetic acid inhibitor. And it's more flexible because of this sp3 carbon here after next to the carboxyl. In contrast, citraconic acid and mesaconic acid here cannot do the same electrostatic interaction because of the rigidity of these molecules. So these are conjugated systems, so they are rigid and cannot adopt the same electrostatic interaction. That's why they showing less inhibition of SDH. It was also questionable if this itaconic acid with a double bond in the molecule next to carbonyl group. So if our medicinal chemistry groups here would say, okay, this could be Michael acceptor compound, which so it reacts by covalent interaction, which could be critical. Okay, so we were questioning if this molecule, the itaconic acid, inhibits the SDH via covalent inhibition or just non-covalent interaction. So we inspected all cysteine residues within the binding site, which is close to the carbon group here, and measured the distances. And the nearest one is at 8 angstrom distance, which is far and not suitable for starting a covalent interaction. So for possible interaction, you need like maybe 4 angstrom, right? But if it's far, then of course this is not far. And with this, it confirms that the molecule inhibits SDH via non-covalent binding. Yes, as I mentioned, they are different in their biological activity. For example, the citraconic acid, it shows it's the only molecule which inhibits the immune responsive gene 1 protein. It's a mitochondrial protein responsible for production of itaconic acid via decarboxylation of this substrate, the cis-aconic acid. So this carboxyl group D removed, and then you get double bond here and isomerization ending to up with itaconic acid. So the ERG1 is responsible for this process, and the only inhibitor of ERG1 is the cycloconic acid. So how to interpret this? We checked with the similarity analysis for these compounds to the substrate, the cycloconic acid. And in fact, actually, the citraconic acid showing the highest Tenmuto coefficient, 0.7, compared to the other molecules. So it could be a substrate, a competitive inhibitor. So it competes with the substrate for binding into the catalytic active site. And with this substrate, this aconic acid cannot bind. Okay, and this was the predicted binding mode of citraconic acid in yellow compared to the substrate in magenta. This is aconic acid, and as you can see, it nicely fits to the binding site. And later on, after definition of the crystal structure, it turns to be the same binding mode. Last example for studying the structure, understanding the molecular mechanism of inhibition of the compounds via docking is the interpretation of the improving cytotoxic activity of the 4-octyl itaconate on the cells. So 4-octyl itaconate presented in this structure, it enhances the oncolytic virotherapy by multi-target inhibition of antiviral immunity. So oncolytic virotherapy. Have you heard about this? So you know the phage therapy. So targeting bacteria via viruses, via phage. It's the same, but applying it on cancer cells. So with special viruses, targeting only the malignant cells, it will enter, replicate, destroy the malignant cells, and not affecting the normal cells, which is very good. That's called oncolytic virotherapy. It was found that 4-octyl itaconate improves this anti-tumor activity of the viruses by inhibiting the immune response against the virus. Okay, by affecting mainly two proteins, IKK beta and MAVS, sorry. Okay, we will talk about MAVS later. So this was done in collaboration with David Oladmir from Denmark. So he sent to me this data. So this is the LCMS analysis of cystinome modification after incubation with this compound. So this compound acts via covalent interaction with the protein, right? Because of the Michael acceptor group. And then you can check which protein was interacting. And he found the IKK beta and other proteins, which is totally fine. Even though for the single protein IKK beta, right, there is a list of 7 cysteine residues which was interacting with 4-octyl itaconate. So how the compound at the end inhibits this IKK beta? Okay, so I started with covalent docking. So there is also approach. You just not make non-covalent interaction, but you can also make covalent interaction, covalent docking of the molecule by irreversibly binding to some reactive groups in the protein. This could be cysteine, threonine, lysine, and so on. Anyway, so I did it only for 6 cysteine residues for which crystal structure, experimental crystal structure was available. And here are the binding energies. Okay, the lower the binding energy value, the better the binding. Excellent. And yes, again, sorry for this misalignment. Anyway, the 3 amino acids here. So the cysteine 179, 412, 464, showing actually the lowest binding energy, which interestingly matches very well with the 3 amino acid residues showing the most amplification or predominance after incubation with the 4-octyl itaconate. So this gives you indication that these results actually could be reliable and you can interpret it. First thing. The second thing, still we don't know how the compound inhibits the protein, inhibits the IKK beta. So first, looking at the cysteine 179, it's located at the phosphorylation binding site. So it's kinase, IKK beta, sorry, it's kinase protein. So kinases make phosphorylation. Okay, so this located at the phosphorylation binding site, and there is a phosphorylation loop here, which is flexible, like opening, blocking the phosphorylation site. And if the molecule for octylitaconate interacts covalently and binds here, it interferes with the movement of this loop. And that's why it blocks the phosphorylation. The protein has another function, which is the protein-protein interaction with other regulatory subunits called NEAM. So here is our protein, the IKK beta, and here is the crystal structure together with the interacting group of NEAM, this alpha protein. And as you can see here, if the molecule binds at the cysteine 716, it actually makes clashes with the two amino acid residues here, isoleucine 71, arginine 75, coming from NEMO. So if the molecule is present, the NEMO cannot bind to its binding partner. So then you can understand how the molecules are inhibiting or affecting the protein function. So again, nothing without a limitation. So we discussed some of them, and the important factors here actually is the data quality. Be careful when you choose your crystal structure you want to work with. Check if it's experimental, if it's high resolution, and relevant to your case. You're working with human proteins. There is also another species. So just check the most relevant protein and so on. And here is actually a nice example from AlphaFold for the mouse. So this mitochondrial antiviral signaling protein. Actually, I refuse to work with it because this is the predicted structure, as you can see how it looks like. It's not tertiary structure. And this is the accuracy or the, again, sorry for this. So it's expected position error. So if how far would be the position of the amino acid compared to the predicted structure. And here's the higher angstrom, of course, the less accuracy, and only small part of the protein showing good prediction. And just to be sure how your protein should look like, this is the prediction accuracy for another protein with mini crystal structures. So of course the prediction would be more reliable. But in this case, please go for another method. Second limitation, protein conformational changes. Whatever the resolution you get or you use for your in silico work, if it's 1 angstrom, it's the highest resolution. Be careful, it's a single snapshot of the protein. It's not the real case. The protein is always dynamic and moving and takes different conformations. Okay, so this means just be careful when you interpret the data at the end. What you can overcome this by making MD simulations, as we know now, but of course this is computationally costing. Okay. Last limitation from my side is the binding site prediction. Since this was a question here about the binding site prediction. So also be careful. There is many programs where you can predict the binding site. No necessity to have a ligand with the protein to locate the binding site. Also for the ABO protein, you can determine the binding site. Okay. Nevertheless, be careful because also the protein and the binding site can change in absence and presence of a ligand. The size of it, the protein adapts itself again in presence of a ligand. Okay, so check this crystal structure. Is it without ligand or with? And yes, be careful. So take home message, highest possible quality of data. Always use highest possible quality of data in terms of the ligand, the library you choose, and the target structures, and also the program you are using. There is many free software. If it's not good, you waste your time. Otherwise, yes, you call it garbage in.
Garbage in, garbage out.
Exactly. Okay. The alternative, forget about in silico screening, silico drug design, and use experimental methods. Okay. So here is an example of actually it's drug repurposing project for in silico screening targeting respiratory syncytial virus, RSV. This was done during the Corona time. So we screened for drug repurposing library composed of 12,000 compounds against the inhibition of the RSV virus. It's virus infecting the lower respiratory tract, mainly affecting children and elderly, and of limited treatments. Okay, and they identified lonafermin. It's a pharmaceutical transferase inhibitor drug approved by EMA and FDA for Hutchinson-Gilford progeria syndrome. Interestingly, that's also rare disease. So it's funny because we took a drug from rare disease and repurposed it for a common disease. Yes, if you got a hit like this, belongs to a certain group of inhibitors like pharmaceutical S-transferase inhibitor, you expect that other S-transferase inhibitor, pharmaceutical transferase inhibitor, should be active also. And we took this compound, tipifarnib, and tested for antiviral activity, but it doesn't show antiviral activity. This means that the mechanism of inhibiting the RSV virus doesn't depend on blocking the pharmaceutical transferase. Okay, it's another mechanism of action because similar compound from the same group doesn't inhibit. And as you can hear here, the antiviral activity, the lonafermin, potent IC50, low CC50 cytotoxicity, and compared to tipifarnib, it's much more potent. And here in the big assay, also no formation of the legs of the virus in case of lonafermin, in contrast to tipifarnib and TBC. How you know the mechanism of action? Via raising the resistant strains to the drug, right? You passages and make gene sequencing and identify the mutations in the virus genome, okay? And this revealed that the most mutations are in the RSV fusion protein. So it's the protein responsible for attachment to the cell and interaction. Great. So then we need to validate this experimentally. And for this, we use surface plasmon resonance, SPR, and compared it to the tipifarnib. So we injected the compound at different doses. And as you can see here, a dose-dependent increase in the response, indicating good binding. It's reproducible. In contrast, at the same concentration, tipifarnib doesn't give significant high response, or also it's dose independent. So confirming that the compound binds to this F protein. Furthermore, this also was confirmed using crystal structure determination. This was in the correction of the paper. So the reviewer were not convinced by SPR and asked for crystallography. Perfect. So to sum up, in silico drug design offers versatile methods of drug rediscovery in a timely and cost-effective manner. Experimental screens are essential anyway for hit identification and validation of on-target effect. You need to validate this, okay, to strengthen your hypothesis or your claims. The best approach to make combination, to start with in silico, define, focus your active compounds, and then test. Instead of testing thousands, you can test the top 100, whatever. Combining both in silico and experimental strategies would save time, money, and reduce attrition rate. So the failure of the project This could be minimized if you combine those. And yes, so the most exciting part, my lovely part. So based on this, we joined expertise between Dr. Lalia and I, and then applied for this project. So new opportunities for developing service for rare genetic diseases. And this is funded by AGIA. Many thanks, Dominic, here to be within us. And yes, so basically, it starts with data collection, the pre-medit platform. Also, we plan to make a database for targets, potential targets, could be investigated, then applying the CAD approaches, and ending with validation using biophysical methods, SPR, MST, and also cell-based assays. For this, yes, I'd like to thank my collaboration partners, my small group, Gabriela, Fled, and Ahmed, also Dr. Lilia again for the kind invitation and the hospitality, AGIA. And if you are interested in AGIA and finished your PhD since 3 years or higher and excited for interdisciplinary work, please be tuned. We have a new call for membership early next year. With this, thank you for your kind attention and apology for more time.
Thank you, Dr. Walid, for this nice presentation. Let's move to questions. So we'll begin with Hussein.
Thank you for the nice presentation. I'm curious about the covalent docking. So you mentioned that you adjust the docking poles to include some reactive groups. I'm wondering about the architecture of the pocket. You will also have some specific properties that you need to cover for the pocket, or it's just a reactive group?
Basically, the active group to start or trigger the reaction, the chemical reaction, but it relies at the end on the binding score. So if the active group is surrounded by hydrophilic surface and your compound is hydrophobic, yes, you will see something attached to the reactive group, but with binding energy of -3 or -2, very low, because there is no possibility of interaction.
So you still need a pocket.
Yes, so actually you need a normal pocket with some hydrophobic residues and nice inclusion into the protein to expect higher binding, possibility of binding. Because actually in most cases, covalent binding occurs after non-covalent interaction. So the molecule would first approach the binding site, binds nicely, and then due to the proximity, it will interact, but not like going with the Michael acceptor and looking for whatever reactive group. So of course, you need a nice, well-defined pocket.
Next question. Okay.
Yeah. Thanks, Dr. Walid, for
My pleasure.
very nice presentation. So I like the example of RNA polymerase
Yes.
and HIV
Reverse transcriptase. Yes.
So yeah, my question is, how did you start this? So you choose to compare both.
Yes.
So based on literature or based on the similarity of the pockets, or so
Yes.
how did you?
Thanks for this. So actually, at the beginning, it was taken from the patient. So where the switch region, the antibacterial target, was defined, it was reported by the group that it's reminiscent to the reverse transcriptase binding site.
Yeah.
And based on this information, we invested more. Okay, so should this molecule can also bind or not? Yes. Thank you.
Thank you.
When we cite the limitations, you said that we need to be careful when choosing the binding site. If it's vegan or not. Isn't it the most rigid part of the structure? So you can't really detect it that way.
No. So basically, in some cases, if you have the crystal structure of a protein without a ligand. You using the software to predict the binding site. It could overlook some binding sites because they are occupied by the protein itself. And it's known, for example, the reverse transcriptase, of HIV. This binding site doesn't exist if the ligand is removed. So there is like a gate, and this gate opens to accommodate the compound, and if the compound is not there, then the gate closes the binding site, and there is no binding site. So you may overlook the binding site. Okay, and of course, of this induced fitting and so on, the protein, if it's flexible, it can also accommodate smaller molecules, larger molecules, so the binding site could vary a bit. So it's better, for example, if you have both with ligand, with no ligands, and check from structural biologists if there is significant difference or not. If not, great, use any of these crystal structure. If there is difference, of course, you need to work with structure with ligands, because you also will introduce a ligand and not the ABO enzyme and the protein.
Okay, go ahead.
Sorry, if there is anyone else who wants to ask a question. I can be the last. I can be the last one.
Okay.
If there is one.
Any more questions? No. So
you mentioned the example of flexibility. I think this could be easy if you have like a hollow and an acoustic protein, then you can maybe make the booking on both of them and check the results later.
For flexibility. For
the flexibility. So now for a protein that does have, let's say, one crystal structure, how could you generate the flexible conformations on which you can do the booking data?
So yes, okay, thank you. So I'll try to answer this. So of course, you know the function of your protein, and it's known for some proteins. It's just structural protein. This means with limited flexibility. This means if it's flexible or not, this have a minimum contribution to the quality of your output. But if the protein is really flexible and dynamic and opens and close, like some cases, then of course you need to choose between the closed conformation, the open conformation, and which one is more relevant. If it's one, okay, maybe you rely on AI to generate another protein, but AI will give you the same, more or less the same crystal structure based on the experimental data. But as I mentioned, my message, don't over-interpret the results you will get from this. But of course, if it's well carefully used, and then it will be also good results. Yes.
No question. We move to Kenan.
Thank you.
I hope so. Here.
Okay, just 1 minute. Okay, can start. Yes. So it's a pleasure to present to you our speaker, Dr. Kenan from Germany. So Dr. Kenan is a distinguished researcher specializing in synthetic biology and bioinformatics. He leads the junior research group on synthetic biology and of microbial natural products at Helmholtz Institute in Germany, and also serves as a CSO and co-founder of Maria Bioscience AG. So he has a PhD from Goethe Institute, Goethe University in Frankfurt. His groundbreaking work on non-ribosomal peptide synthesis has earned him several patents and high-impact publications in several journals, including Nature. So Dr. Kenan has held leadership roles at several institutions, including the Max Planck Institute, and he is the recipient of several awards, including the Helmholtz Young Investigator Group grant. So his research focuses on developing innovative approaches for engineering microbial bioscientific pathways in order to create novel therapeutics, particularly antibiotics. So, Dr. Kainan, please start the stage for you.
Thanks for the kind introduction. Thank you for inviting me and giving me the opportunity to present my work. It's late. I see many tired faces. Now you're facing a totally different topic. So it's not really about repurposing. It's not about medicinal chemistry, but still about drug discovery at least. And I press here. Right? Yeah, so one of the major drivers or motivations of my work is basically contributing to finding a solution to the antibiotic crisis. And as we at the Pasteur Institute, I guess I don't have to introduce this to you, and everyone knows about the escalating numbers and that we're basically facing the threat of reaching 10 million deaths per year in 2050, or potentially even higher numbers if these scenarios are right. Some people say it will not be that dramatic, but it looks like actually we are going this way. And besides AMR, there are many other diseases which we can't properly treat. And tuberculosis alone is killing every single year 1.5 million people. So also these diseases contribute to pathogens that are threatening our lives. And of course, we're looking for solutions. And besides small molecules, which apparently everything here is all about, I don't care about small molecules. I care about natural products, basically. And bacterial natural products not only are structurally diverse, as you can see here, but they're also bioactive because they're evolved by nature for particular targets. And sometimes we are happy to discover them, and they're luckily also targeting pathogens, so we can kill them or we can use them to treat cancer because by accident, because nature did not evolve them to treat our cancer, but the targets are similar, so they can bind and they're highly effective. And even nowadays, where we have all these small molecules and fancy drug pipelines, even today, about 43% of all drugs on the market are derived from nature. And this is particularly even more striking if you look at antibiotics and cancer drugs. So it's the majority, it's 60% of all the drugs that cure us when we are ill from pathogens or from cancer, coming still today from nature. And the natural products I'm interested in are actually non-ribosomal peptides, and they're made by huge mega enzymes. And just maybe to give you a figure, like if I'm the average protein in the PDB, these enzymes are as huge as the Eiffel Tower. So they're really, really big. They're challenging to work with. They're super complex. And they're beautiful, in my opinion. So they're very, very well orchestrated mechanism. They're activating substrates from within the cell. They're modifying them. They're connecting them. And in the end, usually they're also cyclized. And the best about these peptides, they're not the usual standard peptides made of L amino acids. So that's not the proteinogenic ones. But we have more than 400 different building blocks discovered up to now. Which means you can make crazy peptides with D-amino acids, with glycosylations, fatty acids, methylations. You can make microacceptors, whatever. So, and every day we're discovering more and more and more chemistry that chemists actually cannot redo in the lab very often. It's either you waste a total PhD thesis to get the to synthesize such a compound, or you just find them and you don't work with them further, and that's what usually happening nowadays. So when we find cool compounds, people don't take them up, try to develop them into compounds because they're much too complex and too expensive to produce. And most famous examples from these enzymes are actually penicillin. I hope everyone knows it because it saved millions and millions and millions of lives. And another one, very famous one, is cyclosporin, which basically made organ transplants possible. And apparently, they're very well suited for drug discovery, natural products. But as I already said, these compounds have been found like this in nature, that even today, we can't even make penicillin. So the penicillin you get from the pharmacy is not made by chemistry. It's still a semi-synthetic. So like amoxicillin, the new, more active variant, is still today made in several different places all over the world. First, they cultivate the fungus, they purify penicillin, they cleave off this part, they ship it to China, then they attach the hydroxyphenylglycerin that has to be there, then they purify it again, ship it to Europe, they pack it, they distribute it. So it's still today such a small molecule, we don't do it fully synthetically. And because they're so complex and you find it like cyclosporine, if cyclosporine wouldn't be perfect to be used in humans, there's no chance. Yeah, so you can't make hundreds of derivatives and explore the structure-activity relationship. Simply not possible, or too expensive. And even if you could, in the end, you have to produce this compound, and every pharma company will tell you, just too expensive. We don't care. Okay, so apparently the solution is engineering these enzymes, and this is something I'm very interested in, and I was not the first person to come up with engineering these enzymes. They have been identified in the 60s. First successful engineering experiments have been done in the 80s, and then basically nothing happened. So there may be 10 publications from the 80s to up to today where people successfully were able to engineer these enzymes. There have been, in principle, 3 different strategies. And the easiest one, of course, is to introduce point mutations into the active sites to alter the substrate specificity of the domains that select the substrates. Sounds easy. Doesn't work. You can do it in vitro. It works beautifully. But when you put back the domain into the big protein, it's dead. Yeah, so there are different substrate specificities on all these different domains in the enzyme, and they just say, I don't expect this. Gatekeeper, off. That's done. So more disruptive, people try to change domains, people try to change full modules. So one module is always kind of 3 domains in one module, and you have these repetitive units, which you should think that works. Yeah, you change the substrate activating part, the transport arm, and the condensation domain that connects both amino acids, and still doesn't work. And that's where I started. Basically, there was a review when I started my PhD that said this will never work. Very encouraging, I guess. Okay, so what I did is basically structural bioinformatics. So I went into the structures, and I found something very surprising. It was known from literature that you cannot cut in between the condensation and the adulation. Because there was a crystal structure of a full module, it was a science publication, famous. And they said this is a stable workbench. You're never allowed to separate them. So, but what I did, I downloaded all the PDBs. They simply superposed them, and what you see is that the smaller crystals that only had C and A domains, or T and C domains, they change. And the interesting thing is that this part was not associated at all in one crystal structure. It was separated. And then I went into analyzing the interfaces. And this is the only interface in the full assembly line. There's no specific interaction, just hydrophobic patches. So I was like, okay, why not cutting that? Although literature said, don't do it, we did it, it worked, and it was amazing. And we could also then come up with some rules because of the specificity issues. So we said, okay, if you select a module with a certain substrate specificity, you also have to check the condensation domains because they also have it. So we had basically 3 rules, and it worked basically all the time if you adhered to these 3 rules. And at this time, we argued, okay, nature is so diverse and there are so many building blocks. It doesn't matter. These rules, it works. You can just have to do explore the whole genomic diversity, genome mining. But the truth is, nature is not that diverse. If you take proteins from similar organisms, they all produce the same compounds, they use the same building blocks because they evolved in a certain niche, and they use the same weapons. So you have to go to, I don't know, into the Arctic maybe to find a different chemistry. But then these proteins, you cannot fuse them to these proteins. Different GC content, codon usage issues. Yeah, doesn't work. So we had to come up with something else. And to overcome this substrate specificity issue, we again went into crystal structures. We found, okay, actually, this condensation domain, it's not a monomer, it's kind of a dimer or a pseudo-dimer. It is two parts with a linker. Why not just cut? And by cutting them, we actually could separate the specificities of the down and upstream substrate that has to be condensed. And it worked. That was beautiful because the idea was, can you make libraries? That was the intention. We had to be better, we had to be easier. And yeah, just to show you how it in principle works. Let's say you want to make this small peptide consisting of a fatty acid, a proline, alanine, two leucines. What you can do is you can basically draw the enzyme you need. And based on what you need, you can now start with genome mining and look for these building blocks with the right specificities everywhere. Then you identify your biosync gene clusters, you amplify these blocks, you recombine them into a plasmid, you put it into E. coli, and produce it. And this then actually worked. In this case, we got, 8 milligrams. It's not so important, but that's basically how it works. Yeah, just build plain Lego with this system. Okay, as a next step, to further get better, because we were still restricted, we still could only use protein parts, bits and pieces from organisms at least using the same codon usage. Yeah, so it didn't have to be like only gram-negatives, you also could use gram-positive building blocks and could mix them a little bit. It was always challenging. If we tried something crazy, this didn't really work. So we thought, okay, we have to learn how nature evolved this. What did nature do? So we analyzed it using some machine learning and principle and found something super surprising because we were not the first ones. There are many, many studies who tried to figure out the evolution of these proteins. And what they never saw was basically this. And this was within the violation domains. These small transport domains, never ever looked at them because they have no catalytic activity. They do nothing. Yeah? They just transport the intermediates. No one checked them. No one was interested in them. And this was striking. Okay, that's weird. Nature seems to use this domain to recombine and evolve its own enzymes. And when we went into the old data sets from the other publications, we found the same. Everyone, and they also saw it, also in their plots, you actually saw it. They just overlooked it. They didn't care. Yeah, so we didn't find it. We just got aware of this. And based on this, we then also could come up with an evolutionary model. So what we found is basically this is the unit that moves around in evolution on top to change the specificities of the substrate selecting parts. We found, okay, nature also changes this independently of the moving parts. So basically, we have now two parts we can exchange, and this works pretty well, as you should see here. I guess this doesn't mean too much to you. For us, this was major, because we could now recombine genetic fragments from all kinds of different organs, high GC, low GC, gram-positive, negative, fungal origin even. And we could even combine proteins from different origins that not only were making peptides, but also ketides. And so we could fuse two different enzyme families to make hybrid molecules. And then we were kind of, yeah, we are so cool. What can we do? And for publication, if you say, okay, you're doing engineering and engineering, in the end, no one cares anymore because you're just coming up with new methods. You have to show that you can kind of make something useful. So we said, okay, we had a collaboration partner in Munich, and he was an expert in proteasome. So the proteasome is a potential cancer target because you can, if you inhibit it, induce apoptosis. We said, okay, this was our collaboration partner, proteasome is our target. And we got them basically inspired by phenlutamide, which is a known proteasome inhibitor. And said, okay, then let's make a small library of similar compounds. Yeah, so that's what we did. We used the biocombinatorial approach, made a couple of compounds. That's then, together with our collaboration partner, we selected these four. Actually, only one of them was supposed to work properly, according to him. All of them worked. And here's then basically the results. We found a selective inhibitor for the beta subunit in the low micromolar range, which had a similar binding mode to the known inhibitor. For sure, not the next super drug, but it just shows that we can do in the lab in Silico design a biosync gene cluster from 5 different biosync gene clusters from 5 different organisms to produce a novel biosynthetic pathway that produces a compound in bacteria that could potentially be used to treat cancer. So, big issue with this technology is it's tricky when you want to modify the organisms of the biosynthetic gene clusters. Even one module is about the size of 3.5 to 4.5 kb. Who's doing cloning here? Is someone cloning? So basically, if you have 5 modules, you're already at 20,000 base pairs. If you have 10 modules and the average number is 9, you're about 30 to 40,000 base pairs that you have to recombine, put into a plasmid, and transfer into a strain to produce it. Big issue. So if you want to do something super cool and quick, we had to change something. So we had to reduce the complexity of these enzymes. So what we did was, I mean, it's easy, right? You see a huge protein, how to make it simpler to work with it, you disrupt it. You make it smaller somehow. And what we found were basically synthetic zippers. They're known. We didn't invent these synthetic zippers. They're just alpha helical structures with a lot of leucines, and they interact with each other. And within the physiological conditions in a cell, it's basically a covalent bond in the end. So we disrupted the genes for this big protein and encoded them on 2 different expression plasmids together with the sequences for the synthetic zippers. Then you just co-transformed them into E. coli and produced these compounds. And interestingly, basically, we didn't really see a drop in titers. This worked very nicely. And if you see this, of course, the next question is, can I use this to do engineering, to create libraries? And the cool part is, you don't have to do the whole work again. You can use, again, the plasmids you had before, and you just create a new one. And now co-transform these 2, and you produce a new peptide. And actually, quite tricky one. So synthetic chemists are not very happy with this simple cyclic peptide. Okay, and now if you now just imagine, your PhD project is to make 1 million of these enzymes. I'm pretty sure no one would be very happy to do this. By simply cutting them into 2 pieces, you only need 2,000 plasmids. Yeah, 1,000 plasmids A, 1,000 B, together 1 million. Pretty simple. If you now think again about this a bit further, if you could split them into 3, 4, 5, or whatever. So, but with 3, you basically need only 300. And we were able to show that spoiler. Okay, so 300 plasmids, for sure in the PhD thesis, easily doable. And you can make 1 million new compounds. So, we had to test this, of course. Is this actually possible? So we created a first small library. We combined them in all possible ways, found that 8 out of these 11 blocks were functioning together, and created a first small library of peptides. Initially, you see it's kind of some titers are quite nice. Many of them are really, really, really low, easily to detect, but for any application later, horrible. So we had to come up with something to optimize this. And we basically optimized these synthetic zippers. We also have a patent on this. It's basically super stupid, easy work. Just truncate it, elongate it, you introduce flexible parts and so on. A lot of work, but super simple. And in the end, what you see is an increase of 3,200% in titer. So basically from 0.1 milligram to 320, which is an industrially relevant scale, simply by playing around a little bit. And this also worked for the others, not in this impressive way, but still 5-fold, 10-fold increase is easily possible. Okay, making libraries is fun, but if you just do it for academic reasons, it's a nice game. I love it. I don't need more. But actually, if you can't screen for bioactivity, again, useless. You just have compounds. So at this point, we teamed up with a team from the ETH Zurich, and they have a nice microfluidics platform. Basically, and what you do is you encapsulate strains into alginate beads. Very simple. So basically, what we've done, we've put our producer strain together with a sensor strain into one bead and cultivated them. Next, they're growing. And then you can stain these beads, or you already include a fluorescence marker, and you can easily sort them, like in a fax. And then you can pick your hits. So basically, over here it's very easier to see. Here, the red dot is the E. coli strain that produced the compound, and the green ones, the sensor strain, which was in this case Repurposing flowers or gram-positive bacteria, because E. coli is gram-negative, so the screening for compound against gram-positive bacteria died. Here, the peptides were inactive. And to test this, we created 2,500 pathways, expressed them, and we found a series of hits. Of course, not super interesting because it's Repurposing flowers, gram-positive. But for proof of concept, we were pretty amazed. And at this point, we thought, okay, we have enough technology collected, enough patents collected. We start a company. And that's what we did. And at the end of 2021, we together with the ETH technology and our technologies, we created Myria Biosciences in Switzerland. And yeah, that's basically the pipeline. Super simple. We do genome mining. We cut them down into catalytic functional units. We recombine them in high throughput, produce the peptides, and screen against sensor strains. And yeah, so that's basically a summary of what the company does. Is we use these synthetic zippers to create huge libraries at the million scale and screen them for bioactivity. And did not move? No. Did not move again? Nope. It's lagging. Okay. Sorry. Okay, just to show you that if I say millions, that's not a joke. Yeah, because people of course ask, do it. So we found an industry partner from Switzerland, big company, and they said, okay, show it. So we did it. We took one big gene class. I can't show you the real data, so it's all kind of funny in a comic style. Anyways, big gene cluster, 80,000 base pairs. Usually you wouldn't touch it, handling this awful. So we used our technology, we have split them into 3 parts, and did something very easy. We created 500 variants to explore the structure-activity relationship, which was pretty easy because we only had to make them smaller. So from this plasmid, we've cut down this away, and from this side also, and from here, and from here, we made the plasmids just smaller. We combined all of them and screened them for activity. Again, we had 500 variants. We kind of had a very good idea what could work. And based on this knowledge, we created 2.8 million artificial Bayesian gene clusters for screening purposes. And it's moving. Yeah, so what we found in the end is many thousands of hits. The best 1,000 we recultivated and validated that it's actually active. And the 40 best we actually sequenced, because just based on the genomic sequence, we know which structure is produced. Yeah, because we know what you put in, we know what every module does. So simply from the DNA, we can predict what we produce. And then basically now we took these 40 hits, we got them synthesized chemically or purified them from growing cultures and validated them. What I can tell you is, it's okay. They will not be the next drugs. But basically with this knowledge now we can do an iteration, start over again. And the fun part is this only takes a week. Yeah, in a week we can screen 3 million peptides. If you do this for half a year, I'm sure you're much quicker than 15 years of development from the antimicrobial compound, and much cheaper. Okay. With this, I realized we can generate a lot of data. And I wanted to explore AI. And the vision of my work I'm doing now at the Helmholtz is basically to create AlphaBuild. And what AlphaBuild does is only you put in a structure, and at the end, your AI gives you a Bayesian gene cluster that will produce the structure. Yeah? Pretty simple. And if you think about this further, where I want to be, maybe in 10 years, ideally, we don't put in a structure. We put in a target. After we have screened enough targets, created enough data, it will be for sure possible. We give it a target, to get a library of 1 million Bayesian gene clusters, we put them into our screening platform, take the data, and start over. So, but how actually doing this? These are big words. It's tough and expensive. So we start small. Also, this is kind of, these slides are not nice, and actually I'm not really allowed to talk about it, because the patent company of Helmholtz came. And anyway, I think that's good enough. So I can tell it. So my student created basically a high throughput workflow. And what she now does is creating 1,000 novel Bayesian gene clusters producing peptides every single week. And we are not using synthetic zippers. Yeah, so the approach I showed you before is amazing. We can make millions, but the only ones we learn anything about is the ones we can pick out in the screening. The rest is dark matter. You have no clue. This doesn't work. We need high quality data. You need to know the exact genetic information. You need to know the compound. You need to know the bioactivity, so is it active against antimicrobials, and how much is produced. Yeah, so we can now at the moment produce and analyze 1,000 compounds per week in my lab. She's done this now 5 times. You've now basically 5,000 artificial NLPSs in our dataset. It doesn't sound much, but up to today, scientists since the 60s only characterized 10,000. Yeah, it will be at 10,000, I guess, next March, which is basically more what scientists in 60 years of research did. And we made them artificially. And we want to expand this to 1 million. With this data, we're creating a database. It's called the repository of bioengineered gene clusters. And we also collect data from literature. We put this all in and prepare it in a way that it's AI and machine learning ready. On top, what we have in my lab is at the moment basically 200 different modules that we can use to do these experiments. Yeah. That's basically this project to generate data. And right at the moment, the first project started where we use this data to make predictions and to use this data to already design new enzymes. It seems actually to work out even with these small data sets using machine learning. Really like simple things like random forest performs crazy well. Okay, next step. Much cooler. The big question behind this, can we use AI as it is at the moment to produce novel enzymes that are active and do what we want? And to explore this, again, we need the data. So what we do at the moment is, on the one hand, we use simple AI models and ask it to generate for us protein sequences. We do this at the moment about 10 to 50 per month, because we have to synthesize them, that's expensive. It's slow until these genes arrive. It's again, a month is gone. You are spending 10,000 euros. And it's slow. So we started to make lab-scale evolution experiments, basically simply using polymerases that introduce random mutations into our proteins. And here we can easily make 100 proteins per day. So we collect a lot of data. That's pretty simple. And these data basically go into, basically drug design tools. So we are repurposing drug design tools. We're using molecular dynamics, binding free energy calculations to figure out why do these enzymes behave like they behave? And on top, we use drug design to actually design proteins. It's like a rational design approach based on molecular dynamics, and also test them. But again, they are part of this because we have to again synthesize them chemically. That's slow. That's maybe 20 per month. That's not much. In the end, all this data feeds into supervised machine learning, which goes on top of the AI models that conditions the AI to produce good sequences basically from the beginning. Yeah, because at the moment, if you start with 1,000, you test 1,000, you get a couple of functioning ones. And with this very quickly led to, which is really impressive, this is project just half a year old. No, the molecular dynamics is, Anyway, wild-type enzyme. Our best AI-generated enzyme up to date, which outperforms the wild type, basically 1.5-fold. So it produces 50% more of the peptide than the wild type enzyme, which can be very important if you want to produce drugs biosynthetically in the future. But it also, of course, opens doors to create enzymes, functionalities that nature never evolved. I hope. So basically, to come to an end, I guess we create kind of the first cyborg NRPS. Pretty sure we are the first ones, because no one touches these huge enzymes. But it really shows what will be possible with AI, let's say in 10 years. If it's already now possible, if you have a couple of thousand data points, which is not much, but they're high quality, they're made in our lab, we are still constantly going from AI to wet lab, AI, wet lab. Yeah, I'm convinced in a couple of years, this will be very, very powerful. And we just applied for a lot of money to actually create this full alpha build pipeline, because what you realized, it's fun. So, no, yeah, it's fun. It's a lot of fun. It's super expensive. Yeah, just getting all these genes is expensive. Computing, we calculated we will need about 2 million GPU hours at the supercomputing center in Jülich, which is the most powerful supercomputing center in Europe. You need money. Yeah, so, but basically now we have the pipelines, we have all the proof of concepts, and we hope to get the money to actually create this. And now I'm basically, at the end, I have no summary slide, but I still hope I could convince you that not only drug design is fun, protein engineering, protein design is at least the same fun, and you can use it also to create and design novel molecules with bioactivities that nature never invented. And yeah, of course, this is not all my work, because I did not most of it. And so thank you to all the collaborators, to my old group, to my new group, to my company, and for the funding, of course. That's it. Thank you.
Thank you very much, Dr. Kenan, for your insightful presentation. Have you any questions? It was very complicated.
The last presentation.
To synthesize some molecule in your lab.
Sorry?
The technique
yeah.
To synthesize some molecule. It's in your lab or
I mean, we do it
other.
We do it biosynthetically.
Society.
So basically, we do biosynthesis. So we produce all these compounds in our lab, and if we need standards like from chemical synthesis, we go, I don't know, to China, basically.
Okay. So yeah, one question. You talked about so when you generate this kind of peptides, so you are creating your own database, which is called Repurposing.
Yeah,
right?
Local data.
And is it linked to other public databases?
Not yet.
Not yet.
Okay. We are finished with this. We will publish this, but first we want the data and use it on our own, and then we make it.
Yeah,
for sure available.
Another thing. So in your database, so starting from one peptide, you generate several. You have this kind of annotation, which means that all the compounds are coming from the initial
so basically,
something like this.
I mean, it's kind of a graph relation database. So what we have is all these modules that we recombine as a layer with all the substrates from these modules in a different layer. Then all these recombinations, next layer, to the produced compounds in the end, next layer, and then.
I like the way that you think, because it's linked to the mRNA splicing, so the exon and something like this. So what you are doing is really to say
similar without cutting out things,
creating something.
Yeah.
We have a question, please, Dr. Kenan. So among the peptides that you identified and generate, do you think that post-translation modification could increase efficiency of such peptides in
peptides or the enzymes?
Enzymes or peptides.
So what we do for the enzymes is genetic code expansion at the moment. It's very early, so we basically use artificial amino acids to engineer these enzymes to introduce funny amino acids into the active sites that they activate things nature would never accept, on the one hand. And on top, I think this technology is beautiful for discovery, but not for drug development. Right? So you will always need chemists that optimize these compounds further. Otherwise, chemists love weird compounds. And we like to add halogens everywhere. Yeah, that's what nature did not do. So what nature did not invent, I can't use. So we will always need that. I think that's a beautiful platform to generate a lot of data, so that medicinal chemists can, inspired by data, optimize these compounds. But maybe genetic code expansion will one day lead that we don't need chemistry, but I doubt it.
Thank
Thank you very much. More questions?
Are you hiring a company?
Sorry?
Are you hiring?
Sometimes, yes. So we just hired.
Okay. So if no question, we can move to the lunch break.
So you